540 million years ago, the ocean did not look alive. No fish, no coral reefs, no sharks moving through blue water. For most of Earth's history, the sea was quiet in a way that is difficult to imagine now. Soft-bodied organisms drifted slowly across the seafloor beneath oceans that looked, behaved, and even chemically functioned differently from the ones surrounding us today. Then something changed, and in a span so short that geologists still argue over how sudden it really was, the ocean filled with forms of life so strange that some of their fossils were originally reconstructed backward. Eyes appeared, armor appeared, predators appeared. Entire body plans emerged almost at once, many of them unlike anything that would ever exist again. Some creatures looked assembled from unrelated animals. Others were so anatomically unfamiliar that scientists spent decades debating how they even moved. The world that followed was no longer passive. For the first time in Earth's history, life began hunting life on a massive scale. And once that began, evolution accelerated into something violent, experimental, and irreversible. But before we begin, I'm curious, is this your first time here, or have you been down this rabbit hole with us before? Let me know in the comments. By the end of this video, the Cambrian Ocean will no longer feel like the distant beginning of life. It will feel like the moment the modern biological world first opened its eyes. Long before the ocean learned to hunt, before anything swam with intent or watched the water around it, the planet drifted through an age of almost unimaginable stillness. Earth was already ancient then, more than 4 billion years into its history, yet the most complex living structures on its surface were thin microbial films spread across the seafloor like dark living skin. Continents shifted slowly above them, volcanoes rose and vanished, ice advanced across entire hemispheres and retreated again. Through all of it, life remained small, soft, and chemically bound to the surfaces where it formed. For nearly 3 billion years, the ocean belonged to bacteria, not primitive in the sense of failure or incompleteness, but limited by the world they inhabited. These organisms absorbed energy, divided, spread, and adapted with astonishing biochemical sophistication. Yet the sea around them remained almost motionless in spirit. Nothing pursued, nothing fled. There were no jaws closing in darkness, no eyes turning toward movement, no nervous systems mapping the shape of the water. Life responded mostly to chemistry, to gradients of oxygen or sulfur drifting invisibly through the sea. The planet was alive, but only in the quietest possible way. The seafloor of this world was firm beneath thick microbial mats that stretched for enormous distances without interruption. Sediment beneath them remained sealed away from oxygen, layered gently over spans of time, so long that mountain ranges could rise and erode, while the ocean floor above stayed nearly unchanged. Even the currents moved through a biological emptiness, difficult to imagine now, carrying nutrients across waters where almost nothing large enough to notice them yet existed. Then, near the end of the Precambrian, something delicate appeared. The Ediacaran organisms entered the fossil record softly, without armor or teeth or shells, their bodies pressed into ancient sediment like impressions left in wet clay. Dickinsonia rested flat against the seafloor, oval and ribbed, sometimes growing longer than a human body. No confirmed mouth has ever been found. It may have absorbed nutrients directly through its skin while lying motionless beneath dim water rich with microbial life. Nearby stood Charnia, anchored upright like a frond swaying weakly in the current, filter feeding passively from the ocean around it. Sprigana carried something stranger still, a faint asymmetry suggesting a distinguishable front and back, a possible early hint of directional life beginning to emerge from the stillness. Yet these creatures remained slow, soft, and deeply unlike the animals that would follow them. They left behind almost no descendants. Their strange anatomies seemed to belong to a separate experiment in multicellular life, one that flourished briefly and then faded from the planet almost entirely. The ocean of the Ediacaran was not yet an ecosystem of pursuit and escape. These organisms did not reshape the sea around them in the way later animals would. They rested upon it gently, like temporary growths upon the surface of a much older world. Then, around 541 million years ago, the record changes. The Ediacaran organisms vanish from the stone with startling abruptness. In the layers above them appear animals so structurally different, so active, and so anatomically complex that early paleontologists believed the dating had to be wrong. The transition seemed impossible. For billions of years, life had advanced only slowly across the margins of complexity. Then suddenly the ocean filled with bodies built for movement, sensing, feeding, burrowing, swimming. The long microbial silence of Earth was ending. The seafloor still carried the memory of that older world, but its stability was already beginning to fracture beneath new forms of life, pressing into the sediment for the first time. The water remained mostly empty by modern standards, vast stretches of ocean still untouched by complexity, yet something irreversible had entered the planet's chemistry and biology together. The conditions that had restrained life for almost all of Earth's history were loosening, quietly, all at once beneath an unfamiliar sea. The change did not begin with an animal. It began with the ocean itself, with invisible thresholds crossed slowly beneath the surface of the world. For most of Earth's history, the sea had been chemically hostile to complexity. Energy existed, nutrients existed, sunlight fell across the water every day. Yet something essential remained just beyond reach, as though the planet had all the ingredients for a new form of life, but had not yet learned how to combine them. Then, over millions of years, several ancient systems drifted into alignment together. The first shift was oxygen, not atmospheric oxygen alone, but dissolved oxygen moving through shallow seawater in concentrations the planet had rarely sustained before. Earlier oceans contained enough oxygen for microbial life and soft drifting organisms, yet active animals require far more. Muscles capable of sudden movement, nervous systems processing rapid information, bodies large enough to swim against currents. All of these demand enormous cellular energy. Beneath a certain threshold, the mathematics of metabolism collapses inward. Above it, entirely new behaviors become biologically affordable. By the beginning of the Cambrian, shallow marine environments appear to have crossed that threshold for the first time, reaching perhaps a tenth of modern oxygen levels. It was not a fully oxygenated world, but it was enough for tissues that consumed energy rapidly, enough for bodies that moved with intent, enough for the ocean to begin experimenting with speed, pursuit, and scale. At nearly the same time, the planet was still recovering from a much older catastrophe. During the cryogenian glaciations, ice may have spread across almost the entire surface of Earth, sealing oceans beneath frozen darkness for millions of years. When that ice eventually melted, enormous quantities of phosphorus washed from weathered rock into the sea. Phosphorus drifted through the water as one of the fundamental ingredients of living cells, woven into DNA, membranes, and the chemistry of energy transfer itself. The ocean responded with an immense bloom of phytoplankton and microbial productivity. More microscopic life meant more food suspended through the water column, and for the first time, there was enough biological surplus to sustain larger organisms feeding upon smaller ones. The continents were changing as well. Across the surface of the planet, enormous land masses were beginning to fracture apart. Gondwana, vast and ancient, split slowly into new coastlines and shallow inland seas. Warm water spread across continental shelves, flooded by high sea levels, creating broad mineral-rich environments where sunlight could easily penetrate to the seafloor. These shallow marine zones became unusually productive, dense with nutrients carried from eroding rock and stirred continuously by tides and storms. Even the stone beneath the oceans carried evidence of instability. Geologists call it the Great Unconformity, a strange global gap in the rock record, where immense spans of earlier crust appear to have vanished through erosion. Entire layers of continental material were stripped away and carried toward the sea, delivering minerals into marine ecosystems on a planetary scale. The chemistry of the ocean shifted under the weight of this influx, becoming richer, more reactive, more capable of sustaining complicated life. Yet some researchers believe the final ignition was not environmental at all. At some point, perhaps quietly and without spectacle, one organism developed the ability to consume another living animal directly. The moment this happened, the logic of evolution altered. A body that moved slightly faster survived longer. A surface slightly harder to bite became valuable. Sensory structures capable of detecting movement gained immediate advantage. Natural selection, which had once operated slowly across relatively passive organisms, became locked inside an escalating biological competition that no lineage could fully escape. None of these conditions alone appear sufficient. Oxygen without nutrients could not sustain abundance. Nutrients without shallow seas would disperse into emptiness. Predation without energy-rich metabolism could never intensify beyond isolated events. But together they formed something Earth had never assembled before, a narrow intersection of chemistry, geography, climate, and biology that transformed the ocean from a stable microbial world into a system capable of explosive innovation. The sea was ready now, though nothing within it yet understood what readiness meant. The water carried more oxygen than before. Minerals drifted through warm continental shelves. Bodies were beginning to move with purpose. Somewhere beneath the surface, the conditions for a new sensory world had already formed in silence. For billions of years, the ocean existed in darkness without truly knowing it was dark. Light entered the water every day, scattering through the surface in pale shifting bands, but no living thing possessed the machinery to interpret it as image or distance or form. The sea could be touched, tasted, sensed through pressure and chemistry, yet it could not be seen. Bodies drifted through one another's presence almost blindly, guided by currents, dissolved molecules, and faint vibrations passing through water dense with suspended life. Then, around 521 million years ago, the fossil record changes again. Eyes appear, not vague patches of photosensitive tissue, slowly refining over immense spans of time, but complex optical systems already functioning with startling sophistication. Trilobites emerge carrying compound eyes built from calcite crystal lenses, the same mineral that would later harden into limestone cliffs and ancient seabeds. Each lens grew as a precisely oriented crystal structure, aligned carefully enough to reduce optical distortion as light entered the eye. Even in their earliest forms, these were not experimental organs struggling toward usefulness. They were elegant instruments shaped by immediate evolutionary pressure. Across the Cambrian seafloor, trilobites moved beneath shallow water, carrying hundreds or thousands of lenses at once. Some species developed holochroal eyes, dense fields of tightly packed lenses covered beneath a single corneal surface. These eyes provided wide environmental awareness, allowing movement and shadow to be detected from nearly every direction simultaneously. Other species evolved something even stranger, schizochroal eyes, where each individual lens sat separated from the next, each with its own cornea and optical pathway. These widely spaced lenses produced exceptional depth perception, a way of measuring distance through the geometry of incoming light itself. The ocean had crossed into a new sensory reality. Paleontologist Andrew Parker later proposed what became known as the light switch theory, the idea that vision did not emerge as a consequence of the Cambrian explosion, but instead triggered much of it. Once an organism could visually identify another body at a distance, every animal in the water inherited a new problem. Shape became dangerous, movement became detectable. The outline of a body against dim water could suddenly mean pursuit, consumption, or escape. The fossil record preserves an eerie coincidence supporting this idea. The earliest mineralized shells and armored surfaces appear almost exactly alongside the first sophisticated eyes. Hard structures spread rapidly through marine ecosystems. At nearly the same moment, vision becomes widespread. It is difficult to separate the two events. The ocean seems to have recognized visibility and protection simultaneously, as though one innovation demanded the other immediately. Before vision, camouflage had little meaning. Burrowing offered no visual concealment because nothing was watching. Speed existed only as movement, not escape. Armor was unnecessary in a world where bodies rarely targeted one another directly. But once eyes entered the water, these strategies acquired sudden value. Animals that blended into sediment survived more often. Bodies capable of disappearing beneath the seafloor escaped detection. Hard outer surfaces interrupted bites and grasping appendages. Evolution accelerated around the fact of observation itself. Even the architecture of the ocean began changing under this pressure. Organisms spread into new ecological spaces, hiding among sediment, rising into open water, adapting body shapes that disrupted outlines or reflected less light. The sea became layered not only chemically but visually, divided into zones of exposure and concealment. Survival increasingly depended on controlling what could and could not be seen. The trilobite eye remains one of the oldest surviving records of this transition. 500 million years later, some fossil lenses are still transparent enough for researchers to calculate how light once traveled through them. Ancient optical systems remain locked inside stone, preserving the moment the ocean first learned to look at itself. And once that happened, the pace of change sharpened immediately. The sea no longer belonged only to drifting bodies and passive survival. It belonged to detection, reaction, pursuit, and defense. Every exposed surface became information. Every movement became visible. The pressure created by sight spread through the water faster than any single organism could adapt to it, reshaping life into forms increasingly elaborate, increasingly armored, increasingly strange beneath the shallow green light of the Cambrian Sea. Across layers of Cambrian stone, paleontologists sometimes encounter a peculiar horizon filled with fragments no larger than grains of rice. Tiny cones, hollow tubes, curved shards, star-shaped spikes. Scattered through the rock in enormous numbers, they appear almost like debris left behind after some forgotten construction. This thin geological layer became known as the small shelly fauna, and it records one of the quietest yet most profound transformations in the history of life. The moment animals began turning seawater itself into armor. Before this interval, bodies remained overwhelmingly soft. Flesh pressed into sediment, drifted through currents, dissolved after death. But once vision and predation entered the ocean together, softness alone became increasingly dangerous. The response arrived not as complete skeletons or fully formed shells, but as isolated mineralized pieces called sclerites, tiny hard plates embedded across vulnerable surfaces, like scales stitched into living tissue. Some covered worm-like bodies in overlapping rows, resembling chainmail. Others formed scattered spikes projecting outward into the water. Many do not resemble any later animal structure at all. They seem improvised, temporary, produced by evolution at tremendous speed before stable solutions had fully emerged. In the fossil record, they appear suddenly and everywhere, as though the ocean had begun experimenting with geometry itself. The Cambrian Sea made this possible through chemistry. Its waters contained unusually high concentrations of dissolved calcium, far richer than most oceans today. Calcium carbonate and calcium phosphate drifted through shallow marine environments in such abundance that extracting them became metabolically inexpensive. Organisms no longer needed to invest enormous energy into producing hard parts. The ocean was already saturated with raw material, waiting to be assembled into biological form. This process, biomineralization, altered life permanently. Cells learned to pull dissolved minerals from seawater and arrange them into rigid structures, shaping armor from the chemistry surrounding them. Every skeleton that would later walk across land, every reef rising beneath tropical waves, every tooth, spine, antler, shell, and bone traces its ancestry back to this transition. The ability to harden tissue into structure became one of evolution's defining inventions. The speed of the change is visible directly in the rocks. Within only a few million years, the seafloor transformed from a relatively smooth microbial landscape into a densely textured environment crowded with mineralized surfaces. Tubes protruded from sediment. Spines projected upward from soft organisms resting along the bottom. Small shelled creatures crept through narrow spaces between encrusted surfaces. The ocean floor developed physical complexity it had never possessed before. Yet the earliest hard parts were often strangely impractical. Many curved outward in elaborate spirals that offered little obvious protection. Some formed delicate stars or branching spicules, too fragile to stop a determined predator. Others coated bodies unevenly, leaving large sections exposed. Natural selection had not yet refined these structures into recognizable defensive systems. Evolution was still exploring possibilities, producing every configuration it could generate within the chemical abundance of the Cambrian Sea. The fossil beds preserving these organisms resemble the aftermath of uncontrolled experimentation. Minerals appear arranged across body surfaces in nearly every imaginable pattern. Long, narrow tubes sit beside flattened plates. Needle-like spikes lie tangled among tiny coiled cones. The ocean was not yet building efficiently. It was discovering what hardness could become. This mineral revolution changed more than survival. Hard surfaces altered movement, posture, and interaction with sediment itself. Bodies gained leverage against the seafloor. Some organisms anchored themselves upright into currents. Others carried rigid supports, allowing them to rise slightly above the substrate. The physical architecture of marine ecosystems became more layered and mechanically complex with each passing interval of Cambrian time. Even death changed under this new chemistry. Soft organisms had once vanished almost completely after decomposition, leaving only rare impressions behind. Mineralized bodies persisted, fragments accumulated in sediment. Entire communities began entering the fossil record in far greater detail, because parts of them could now survive long after the organisms themselves had disappeared. And while the seafloor thickened with plates and spines and mineral debris, another transformation was unfolding in the water above. Bodies were becoming faster there, larger, more directional. Armor had begun spreading across the ocean floor, but the forces driving its creation were already moving through open water with increasing precision and speed. The first fragments were pulled from stone separately, years apart, each one so strange that no scientist imagined they belonged to the same creature. A curved appendage surfaced first in the late 19th century and was described as the tail of a shrimp. Decades later, a circular fossil lined with hard plates was identified as a jellyfish. Elsewhere, flattened impressions in Burgess Shale rock were catalogued as sponges. The Cambrian Ocean had produced an animal so unfamiliar that paleontology spent almost a century accidentally disassembling it. Its eventual name became Anomalocaris canadensis, the abnormal shrimp from Canada, though by the time its anatomy was finally understood, almost nothing about it resembled a shrimp at all. In 1985, Harry Whittington and his colleagues at Cambridge University reconstructed the scattered fossils together into a single body. The shrimp tail became grasping appendages. The jellyfish became a mouth. The sponge became the torso of a large swimming predator, unlike anything then known from such ancient seas. The realization forced scientists to reconsider the Cambrian Ocean entirely. The early marine world was no longer populated only by slow armored crawlers and drifting filter feeders. Something powerful had already evolved there, moving through open water with speed and sensory precision. Anomalocaris reached at least a meter in length, possibly more in some species, making it enormous by Cambrian standards. Most surrounding organisms measured only centimeters across. Against them, its body must have appeared immense, gliding above the seafloor with broad lateral lobes rippling in sequence along each side. These flexible fins undulated through the water in smooth waves, producing efficient propulsion remarkably similar to the swimming mechanics of modern rays and cuttlefish. Unlike many earlier Cambrian animals bound closely to the sediment, Anomalocaris occupied the water column itself. At the front of its body extended two long segmented appendages lined with inward-pointing spines. They curved toward one another like articulated arms, capable of seizing soft prey or pinning armored organisms against the mouth beneath. That mouth formed a circular structure composed of overlapping mineralized plates arranged in a ring, resembling the segmented geometry of a pineapple slice. 32 plates interlocked around the opening, flexing inward as prey was crushed and consumed. For years, paleontologists debated whether the mouth was actually strong enough to break trilobite shells. Then the evidence appeared directly in the fossils themselves. Trilobites from Cambrian rock layers were discovered bearing crescent-shaped bite marks, whose diameter and spacing matched the mouth structure of Anomalocaris with remarkable precision. The injuries survived in stone for more than 500 million years, recording moments of predation from oceans long vanished. Its vision may have been even more extraordinary than its feeding apparatus. Exceptionally preserved fossils from Canadian and Chinese Cambrian deposits revealed large, stalked compound eyes containing as many as 16,000 individual lenses. This gave Anomalocaris visual acuity comparable in some respects to modern dragonflies, allowing it to detect movement, contrast, and body outlines with exceptional sensitivity in shallow marine water. The ocean had already produced a predator capable of actively tracking prey visually across open space. And Anomalocaris was not unique. It belonged to a broader lineage, now known as the radiodonts, an early branch of large predatory animals whose forms varied dramatically. Herdia victoria carried an enormous flattened head carapace, projecting outward like the prow of a ship. Peytoia nathorsti possessed differently shaped feeding structures, adapted to alternative prey. Amplectobelua developed grasping appendages with spine arrangements unlike either of the others. The Cambrian Sea was already diversifying apex predators into specialized ecological roles. These animals transformed marine ecosystems in ways difficult to overstate. Earlier evolutionary pressures had been local and relatively passive, shaped mostly by chemistry and environmental tolerance. Now survival depended increasingly on reaction time, concealment, armor, mobility, and awareness. Organisms incapable of adapting to active pursuit disappeared from ecological space with increasing speed. For the first time in Earth's history, the ocean contained creatures large enough to dominate other animals directly, fast enough to close distance intentionally, and visually sophisticated enough to choose targets before striking. Predation had become organized, directional, and persistent. The sea no longer functioned merely as an environment where life existed. It had become a landscape of hunters and hunted, shaped continuously by the presence of something watching from the water above. High in the Canadian Rockies, where thin air moves across cold stone and snow lingers along broken ridges, there is a layer of dark shale that was once part of a warm Cambrian seafloor. More than 2,000 meters above modern sea level, surrounded now by mountains shaped through immense tectonic pressure, lies one of the least likely fossil deposits on Earth. The animals preserved there lived beneath shallow tropical water 500 million years ago. Their remains reached the sky only because continents themselves refused to stay still. The place became known as the Burgess Shale. In 1909, paleontologist Charles Walcott discovered the deposit in British Columbia while searching for Cambrian fossils along a mountain pass. What he found exceeded anything paleontology had previously encountered. Most fossils preserve only hard structures, shells, teeth, bones, or thick mineralized surfaces capable of surviving decay. Soft tissue almost always disappears within days after death, consumed by bacteria, dissolved chemically, or scattered by scavengers before sediment can bury it. But the Burgess Shale preserved the impossible. Muscles remained visible there. Digestive tracts survived, compressed into thin carbon films. Delicate appendages, eyes, gills, and traces of nervous tissue endured inside the rock long after the organisms themselves vanished. Entire bodies lay flattened into shale with astonishing detail, as though the Cambrian Ocean had briefly learned how to remember flesh instead of merely skeleton. The mechanism behind this preservation appears almost accidental. Around 508 million years ago, communities of marine animals lived near the base of steep underwater reef walls in oxygen-poor basins. Periodically, dense underwater mudslides collapsed from above and surged downslope, burying everything beneath extremely fine sediment within moments. The mud sealed organisms away from circulating oxygen and slowed bacterial activity so dramatically that soft tissues mineralized before decomposition could erase them. Without these conditions, the Cambrian would appear far simpler than it truly was. Paleontology would know only shells, spines, and scattered hard fragments. The strange anatomies moving through those oceans would remain almost invisible. The Burgess Shale became a geological interruption in normal decay, a rare place where time failed to fully dismantle what life had built. Walcott collected more than 65,000 specimens from the site across years of excavation, carrying them back to the Smithsonian Institution and attempting to organize them within known biological categories. Shrimp were identified here, worms there, jellyfish elsewhere. He interpreted the Cambrian through the lens of the modern world because nothing else seemed possible. The idea that entirely unfamiliar body plans could have existed so early in animal history remain difficult to accept. As a result, much of the true strangeness hidden within the Burgess fossils went unnoticed for decades. In the 1970s and 1980s, paleontologist Harry Whittington and his students Simon Conway Morris and Derek Briggs reopened Walcott's collections at Cambridge University and began studying them, specimen by specimen, from the beginning. Freed from the assumption that Cambrian organisms must resemble modern animals, they discovered bodies unlike anything alive today. Limbs appeared where none had been expected, mouths faced backward, spines projected from impossible geometries. Entire anatomical structures resisted clean classification. The Cambrian Ocean emerging from these fossils no longer resembled a primitive version of the modern sea. It appeared instead as a period of astonishing biological experimentation, crowded with forms evolution would later abandon completely. Other discoveries expanded the picture further. In Yunnan Province, China, the Chengjiang biota revealed an even older Cambrian ecosystem preserved in remarkable detail. Greenland's Sirius Passet deposit added additional organisms from polar marine environments. More recently, the Xinjiang biota uncovered another extraordinary assemblage of soft-bodied animals still being analyzed today, preserving tissues and species absent even from Burgess itself. Together, these fossil windows altered the history of animal life profoundly. They revealed that the Cambrian Ocean was not populated merely by familiar ancestors in simpler forms, but by organisms so anatomically unfamiliar that even experienced scientists struggled to understand what they were seeing. Stone preserved them faithfully, yet comprehension arrived slowly, as though the modern mind required time to adjust to a world where evolution still seemed undecided about what an animal could become. Some Cambrian animals appeared strange because they belonged to body plans unlike those alive today. Hallucigenia seemed stranger because even after scientists uncovered its fossils, no one could determine which side was supposed to face the ground. Its body entered paleontology in fragments, pressed into Burgess shale rock, thin enough that details blurred easily into the surrounding stone. When researchers first reconstructed the animal in the 1970s, they produced something almost surreal: a narrow tube balanced awkwardly atop rigid spikes, with soft tentacles waving upward into the water like grass growing from a row of stilts. The interpretation looked improbable even then, yet there was no obvious alternative. The fossils appeared to support it. What were actually soft walking limbs were interpreted as feeding tentacles. What were actually defensive spines became legs. The presumed head sat where the tail truly belonged. The creature received the name Hallucigenia sparsa because its anatomy seemed hallucinatory, almost resistant to coherent interpretation. For years afterward, museum reconstructions displayed the animal exactly this way. Visitors walked past models of Hallucigenia standing precariously on long spikes while its true limbs floated uselessly above its body. The Cambrian Ocean already contained animals unlike anything familiar, and Hallucigenia seemed to push that unfamiliarity beyond biological logic entirely. Later, research corrected part of the mistake. Paleontologists realized the spines could not function as walking limbs at all. The animal was turned upright, placing the soft appendages against the substrate where they belonged. Yet even then, the reconstruction remained partially wrong. One end of the fossil preserved a swollen dark smear that researchers interpreted as a head region, and so the body orientation stayed reversed for years longer. Only later did scientists recognize that the dark structure was not anatomy at all. It was decay fluid forced outward during fossil compression, squeezed from the opposite end of the body as sediment flattened the organism into shale. The presumed head was effectively a stain produced by decomposition itself. By the early 21st century, electron microscopy finally resolved the puzzle in much finer detail. Hallucigenia emerged as a small worm-like animal, ranging from roughly half a centimeter to several centimeters in length, walking across the Cambrian seafloor on paired lobopods, soft, stubby limbs lacking true joints. Along its back projected rigid hollow spines, arranged in rows, defensive structures likely discouraging predators from swallowing it easily. At the front sat a small, distinct head with a circular mouth surrounded by fine feeding structures, almost invisible in earlier fossils. Then another discovery deepened its importance far beyond its strange appearance. In 2015, detailed analysis revealed that Hallucigenia possessed a throat lined internally with tiny recurved teeth arranged in a spiral. These pharyngeal teeth represent the earliest known example of teeth inside the digestive tract itself. They likely prevented food from slipping backward once swallowed, guiding organic material deeper into the body with each muscular contraction. That anatomical detail connected Hallucigenia to a much broader evolutionary lineage. The animal belonged to an extinct group called the Lobopodians, soft-limbed organisms now understood to occupy a critical position near the ancestry of modern velvet worms and distant relatives of all arthropods. Similar tooth-lined, feeding canals persist in modified forms among living arthropods today, linking this peculiar Cambrian creature to crabs, insects, spiders, and countless later descendants spread across Earth. Its body, therefore, represented not evolutionary absurdity, but experimentation near the base of one of the most successful animal lineages in history. Yet the pathway connecting Hallucigenia to that lineage remained so unfamiliar that paleontology required 3 separate reconstructions, decades of reinterpretation, and microscopy technology unavailable to earlier generations before its anatomy could be understood with confidence. There is something revealing in that difficulty. Human perception evolved within ecosystems shaped by later forms of animal life, bodies refined through hundreds of millions of years of selection into familiar patterns of symmetry and function. Hallucigenia emerged from a period before many of those patterns stabilized. Its anatomy belongs to an interval when evolution still seemed willing to attempt structures that later oceans would abandon entirely. Even so, Hallucigenia was only confusing. The next Cambrian organism challenged not merely anatomical interpretation, but the limits of biological expectation itself. When its reconstruction was first presented publicly, professional paleontologists responded not with argument or disbelief, but with laughter rising instinctively through the lecture hall. In 1972, Harry Whittington stood before a room of paleontologists and revealed his reconstruction of a Cambrian animal from the Burgess Shale. The response was immediate. Laughter spread through the audience, not cruelly, but involuntarily, the kind that escapes when the mind encounters something so unexpected that it briefly refuses to process it as real. The fossil looked less like an ancient organism than an improbable sketch assembled from unrelated parts. The animal was Opabinia regalis. Even among the strange anatomies emerging from Cambrian rock, Opabinia occupied a category almost entirely its own. Its body stretched only a few centimeters long, soft and segmented, with broad lateral lobes running down each side that likely rippled gently through the water while it swam close to the seafloor. At the rear extended a fan-shaped tail, delicate and symmetrical, capable of stabilizing movement through shifting currents. But it was the head that unsettled nearly everyone who saw it. 5 compound eyes sat arranged across the top like a row of raised lenses watching in different directions simultaneously. No living animal possesses 5 eyes configured this way. Each appears to have been functional, receiving visual information independently and transmitting it into a nervous system compact enough to fit inside a body barely larger than a finger. The amount of sensory processing required for such an arrangement suggests a level of neural complexity unexpected in creatures from so early in animal history. Beneath the eyes projected the structure that made the reconstruction seem almost absurd: a long, flexible proboscis extending forward from the front of the head like a soft, articulated hose. At its tip sat a small, grasping claw. The appendage appears to have functioned as a precision feeding instrument, probing through soft sediment and reaching into narrow spaces inaccessible to the rest of the body. Its feeding anatomy became stranger still when researchers identified the location of the mouth. The mouth did not face forward beneath the proboscis. It pointed backward beneath the body toward the tail. To eat, Opabinia likely extended its claw-tipped appendage into sediment, seized organic material or small buried animals, then folded the proboscis downward and backward beneath itself to transfer food into a mouth positioned in the opposite direction. The entire process feels mechanically awkward from a modern perspective, yet the anatomy persisted successfully enough to survive within Cambrian ecosystems. Only around 20 confirmed specimens of Opabinia have ever been discovered, all from the Burgess Shale itself. This rarity gives the animal an almost dreamlike quality in paleontology, a creature known from only a handful of impressions pressed into dark stone high in the mountains. Yet those impressions preserve enough detail to place it within a significant evolutionary position. Opabinia is now understood as a close relative of the radiodonts and part of the broader stem group leading toward arthropods. Somewhere deep within its anatomy lies the beginning of a lineage that would eventually produce crabs crossing beaches, spiders moving through forests, butterflies drifting above flowers, and insects filling nearly every environment on Earth. The branch leading toward all of them once passed through forms that looked something like this: soft-bodied swimmers carrying 5 eyes and backward-facing mouths through Cambrian water. Its body reveals how experimental that period still was. Evolution had not yet narrowed animal architecture into the more stable arrangements familiar today. Sensory organs could proliferate in unusual numbers. Feeding structures could detach entirely from the mouth itself. Swimming appendages could resemble flexible flaps rather than true limbs. The Cambrian Ocean tolerated these anatomical possibilities because ecological roles remained open and competition still rewarded unconventional solutions. Opabinia moved through this world not as a failed organism, but as a functioning participant within a rapidly diversifying ecosystem. It searched sediment, processed visual information from multiple directions, maneuvered through the water column, and survived well enough for its lineage to leave descendants stretching invisibly into the future. Yet its body also carried the mark of a transitional age, when evolution seemed willing to test forms later oceans would abandon. Opabinia appeared strange because its architecture sat near the edge of what animal life briefly explored before narrowing again. The organisms that followed would push even further into those abandoned possibilities, building entire body plans unlike anything that survived beyond the Cambrian Sea. Beneath the shallow light of the Cambrian Sea, evolution seemed less like a gradual sculptor and more like a restless inventor covering a work table with unfinished designs. Some lineages would persist for hundreds of millions of years and spread into nearly every ecosystem on Earth. Others appeared briefly, left only a scattering of fossils, and vanished so completely that modern biology still struggles to place them anywhere within the tree of life. The Cambrian Ocean was not merely generating ancestors, it was generating possibilities. Among the strangest were the vetelicolians, animals shaped almost like drifting sacks, with segmented tails trailing behind them. Their front sections were swollen and perforated by a series of openings that may have functioned as primitive gill slits, allowing water to pass through while food particles were filtered from the current. Behind this inflated front body extended a narrower, stiffened tail used for swimming. The overall architecture resembles no living organism cleanly. Some researchers interpret vetelicolians as early deuterostomes, distant relatives of the lineage that would eventually produce vertebrates, including humans. Others argue they belong entirely outside known groups, remnants of a body plan that left no descendants at all. Nearby in the water column moved Nectocaris pteryx, another organism whose meaning shifted repeatedly as new fossils appeared. It was initially reconstructed as a primitive arthropod, but later analysis of dozens of specimens revealed something very different. Nectocaris possessed a flattened body with broad swimming fins, 2 tentacles extending from the head, and what appears to have been a funnel capable of directing water for propulsion. It now seems likely that this animal represented an early cephalopod, an ancestor of squid and octopus already experimenting with jet-driven swimming tens of millions of years earlier than scientists once believed possible. Its reinterpretation revealed something unsettling about the Cambrian fossil record itself. These animals are not simply unfamiliar. They are so anatomically distant from modern expectations that even trained paleontologists can misunderstand them for decades while looking directly at their preserved bodies. Then there was Eldonia, a drifting disc-shaped organism suspended through the water like a soft, translucent medusa. Its circular body enclosed coiled, internal structures whose function remains uncertain even now. Some classifications place it among sea cucumber relatives. Others interpret it as a cnidarian. Still others suggest it may represent an entirely independent lineage with no surviving equivalent in any modern ocean. Eldonia exists in paleontology almost as a question preserved in stone, a body whose evolutionary context dissolved long before humans arrived to study it. Anchored quietly to the seafloor stood Dinomyscus, an organism shaped like a narrow stalk supporting a goblet at its summit. Around the rim extended a crown of delicate tentacles, gathering suspended particles from the surrounding water. It likely lived as a filter feeder, motionless except for the slow movement of current passing through its feeding structures. Yet, despite its apparent simplicity, its biological relationships remain unresolved. No modern group comfortably contains it. What these organisms reveal is the difference between diversity and disparity. Modern oceans contain millions of species, but most belong to a relatively small number of major body plans, repeated and modified across evolutionary time. Fish differ from octopus, crabs differ from jellyfish, yet the underlying architectural categories are limited. The Cambrian Ocean may have contained fewer total species than the modern world, but it possessed extraordinary disparity, a vast range of fundamentally different anatomical designs existing simultaneously before natural selection narrowed the possibilities. Some body plans endured, others vanished completely. Paleontologist Stephen Jay Gould argued that this survival was not necessarily a reflection of superiority. The winners of the Cambrian were not always the most efficient or inevitable designs. They were often the fortunate ones, lineages surviving environmental shifts, extinctions, and ecological changes through contingency as much as adaptation. Replay the Cambrian under slightly altered conditions, and the future of animal life might look entirely different. The fossils preserve this uncertainty with remarkable honesty. Each strange anatomy records an attempt by evolution to solve the problems of movement, feeding, sensation, and survival in ways later oceans would abandon. The Cambrian Sea was crowded not only with ancestors, but with alternatives. And all of these organisms moved through a world still physically unfamiliar compared to any modern ocean, a planet whose seafloor, chemistry, and water itself shaped every experiment unfolding within it. For most of Earth's history, the seafloor remained largely untouched beneath the water above it. Sediment settled slowly through the ocean and came to rest beneath thick microbial coverings that spread across the bottom like dark, living fabric. Researchers sometimes describe this ancient environment as a mat world, a place where bacteria formed dense, leathery surfaces stretching across enormous distances with little interruption. Beneath those mats, the sediment remained chemically stable and largely without oxygen, layered quietly through spans of time so long that entire evolutionary eras passed without the seafloor being physically disturbed. Then animals began digging into it. The transformation appears in Cambrian rock as trace fossils rather than bodies themselves. Narrow tunnels curve through ancient sediment. Feeding trails loop across preserved surfaces. Burrows descend vertically into layers that had once remained sealed away beneath microbial crusts. These structures, called ichnofossils, record behavior directly, preserving movement and activity even when the organisms responsible vanished completely after death. The act itself seems simple, a small animal pushing through soft sediment, yet the consequences altered the ocean profoundly. For billions of years, bacterial mats had stabilized the seafloor chemically and physically. Once burrowing organisms penetrated those surfaces, oxygen-rich water began mixing downward into previously isolated sediment. Organic material trapped below the microbial layer became accessible to deposit feeders searching for nutrients beneath the surface. The old chemical boundaries maintaining the mat world started to collapse under constant biological disturbance. The sediment became alive internally for the first time. As animals tunneled downward and sideways through the sea floor, they created entirely new ecological spaces. Burrows provided shelter from visual predators moving overhead. Some became ambush sites where hidden organisms waited beneath the sediment for prey to approach. Others likely functioned as nesting chambers or temporary refuges from shifting environmental conditions. The Cambrian seafloor was no longer a flat biological surface. It had developed depth, layering, and internal structure. Nutrients locked within buried sediment also began cycling back upward into the water column more efficiently. Each burrow stirred mineral-rich particles and organic matter into contact with oxygenated water, accelerating decomposition and recycling. Productivity increased not simply because more organisms existed, but because animals themselves were physically reshaping the chemistry of their environment through movement alone. At the same time, another transformation rose upward from the substrate itself. The first reefs on Earth began appearing across Cambrian shallow seas, not built by coral, which had not yet evolved, but by organisms called archaeocyaths. These strange creatures resembled porous stone goblets or double-walled vases anchored permanently to the sea floor. Water passed continuously through their perforated bodies as they filtered microscopic particles from surrounding currents. Individually, they were modest structures, but together they formed extensive reef systems spreading across warm continental shelves. Some Archaeocyath reefs rose several meters high, creating the first substantial three-dimensional habitats in marine history. Cavities formed between rigid structures, sediment accumulated in sheltered pockets. Small animals gathered among openings protected from open water predators. The seafloor acquired architecture, vertical surfaces, and hidden spaces where organisms could specialize and coexist in ways previously impossible. These reefs changed water flow as well. Currents slowed around their structures, trapping organic particles and increasing local productivity. Life concentrated densely around them. The Cambrian ocean floor became increasingly irregular and textured, covered not merely in organisms, but in ecosystems, engineering the environment around themselves. Yet the Archaeocyaths themselves remained temporary. By the Middle Cambrian, many reef systems collapsed and disappeared almost as abruptly as they had emerged. One leading explanation involves changing ocean chemistry. Earlier, Cambrian seas contained unusually abundant dissolved calcium, allowing reef builders to construct mineralized walls with relatively low metabolic cost. As calcium concentrations declined, maintaining those structures became more energetically difficult. The first reefs on Earth lasted only around 15 million years before fading from the oceans almost entirely. Still, the transformation they initiated endured. The flat microbial plains of the Precambrian were gone. In their place stretched a seafloor filled with burrows, mineralized structures, feeding traces, cavities, and layered ecological zones shaped continuously by animal activity. Sediment no longer rested quietly beneath the sea. It was being churned, oxygenated, inhabited, and rebuilt from within. And above this newly transformed bottom, the water column itself was beginning to fill with life moving through open space in ever greater numbers, complexity, and motion. For billions of years, the open water between the surface and the seafloor remained strangely vacant. Sunlight filtered downward through enormous volumes of ocean, containing little more than bacteria, drifting nutrients, and microscopic plankton suspended in slow circulation. Life clung mostly to surfaces then, pressed against sediment or microbial mats below. The water column itself existed largely as empty space. During the Cambrian, that emptiness disappeared. Animals began occupying the ocean vertically for the first time, not only crawling across the bottom but swimming, drifting, filtering, hunting, and reproducing throughout the water above it. The sea developed layers of activity extending from the illuminated surface into darker depths below. Motion entered regions that had once belonged almost entirely to currents and suspended particles. Near the surface, dense populations of cyanobacteria and early algae gathered within sunlit water rich with dissolved nutrients. Their photosynthesis tinted parts of the Cambrian Ocean green rather than the deep blue associated with many modern open seas. This upper layer formed the energetic foundation for everything beneath it, converting sunlight into organic material continuously across shallow continental shelves. Within this productive zone drifted the first substantial communities of zooplankton. Tiny crustacean-like animals moved through the water alongside transparent predators called chetonaths, or arrowworms, whose descendants still survive in modern oceans with remarkably little anatomical change. Even in the Cambrian, they already possessed streamlined bodies and grasping spines around the mouth, allowing them to seize smaller drifting organisms with sudden precision. Many larger animals also began life suspended in the water column. Larval stages of bottom-dwelling species drifted freely for part of their development, before eventually settling onto the seafloor. The ocean was no longer organized only horizontally Across sediment and reefs, it had become vertically inhabited. Through the middle depths swam increasing numbers of soft-bodied animals, nearly invisible against the surrounding water. Transparency emerged as one of the most efficient survival strategies available in this new visual ecosystem. Once predators evolved sophisticated eyes, reducing visible outline became immediately valuable. A transparent body required no mineral investment, no heavy armor, no rapid escape. It simply removed the contrast predators depended upon. Many Cambrian zooplankton were almost certainly nearly transparent, much like countless small marine organisms alive today. Invisibility drifted through the water as a passive form of defense, turning the ocean itself into camouflage. Deeper below moved the larger hunters. Radiodonts like Anomalocaris cruised through dimmer layers, processing visual information through immense compound eyes, detecting movement and selecting prey against shifting gradients of light. Their bodies no longer depended on contact with the seafloor. They occupied open water directly, swimming through ecosystems suspended entirely within the sea itself. And below them all, the transformed Cambrian bottom stretched across shallow continental shelves in remarkable complexity. Trilobites walked across mineralized sediment between sponge colonies and reef structures. Burrowing worms moved beneath the substrate. Shell fragments accumulated among feeding traces, and microbial films disturbed constantly by animal movement. The ocean floor had become crowded, layered, and active beneath the drifting world above. As these ecosystems intensified, another planetary process quietly strengthened alongside them. Animals feeding near the surface produced waste and bodies rich in organic carbon. When they died or defecated, this material sank slowly downward through the water column and settled onto the deeper seafloor. Carbon that had once circulated near the surface became buried within marine sediment instead. This process, now called the biological pump, began reshaping Earth's atmosphere and climate over geological time. Ocean life itself started regulating planetary chemistry by transporting carbon downward through the sea. The Cambrian Ocean was not merely supporting complex ecosystems anymore. It was beginning to alter the long-term environmental stability of the planet through biological activity alone. The vertical structure of the sea had therefore become something entirely new, a layered world of production, predation, concealment, feeding, decay, and recycling extending from bright surface waters into darker zones below. The ocean no longer functioned simply as habitat. It had become a three-dimensional biological engine, operating continuously through depth and motion. And among the countless drifting bodies now suspended through that ancient water, there existed a lineage so small and unremarkable that it would have been almost impossible to notice against the surrounding life. Its body lacked armor, speed, and obvious dominance. Yet within it rested an anatomical pattern that would survive everything the Cambrian Ocean could impose upon it. In the middle of the Cambrian Ocean, surrounded by armored shells, drifting predators, and forests of sensory appendages, there moved a small animal with almost nothing about it that suggested permanence. It lacked mineral armor. It lacked crushing jaws, compound eyes, and defensive spines. Against the crowded complexity of the Cambrian Sea, it would have seemed nearly invisible, another soft body passing briefly through ancient water before disappearing into the food web around it. Its name was Picaia gracilens. The animal measured only around 5 centimeters long and swam weakly through the waters preserved in the Burgess Shale deposits approximately 508 million years ago. Its body resembled a narrow ribbon tapering gently toward the tail, flexible enough to undulate through the water in slow coordinated waves. For decades, paleontologists regarded it as the earliest known chordate, part of the ancestral lineage from which all vertebrates would eventually emerge. What made Picaia extraordinary was hidden beneath its soft anatomy. Running along the length of its body was a thin internal rod called a notochord. It functioned as structural support, stiff enough to resist collapse during movement while remaining flexible enough for swimming. This simple feature became the evolutionary precursor to the vertebral column. Long before bones appeared, before jaws, fins, limbs, or skulls, the essential architecture of the vertebrate body had already emerged in this fragile swimmer drifting through Cambrian currents. Along either side of the body lay segmented muscle blocks called myomeres. They contracted sequentially from head to tail, producing a traveling wave of motion through the body wall. Every fish alive today still swims through this same muscular arrangement. The mechanics first visible in Picaia continue moving through sharks, salmon, eels, whales, and dolphins across oceans 500 million years later. There was also a distinct head end, modest but recognizable, establishing directional movement through the environment. Sensory structures and feeding anatomy concentrated at one side of the body rather than being distributed evenly throughout it. The bilateral organization familiar in vertebrate animals had already begun taking shape. These features together formed a blueprint rather than a finished design. Every amphibian, reptile, bird, and mammal that would later exist represents a modification of this fundamental arrangement, stretched, reinforced, specialized, and elaborated across geological time. Yet in the Cambrian Ocean, the blueprint remained delicate and incomplete. Later discoveries in the Chengjiang deposits of China pushed the history of vertebrates even deeper into the Cambrian. Hycoichthys urcaicunensis, preserved from approximately 518 million years ago, possessed a defined head region, sensory organs, a notochord, and possibly one of the earliest primitive skull structures yet identified. Its body measured only around 2.5 centimeters long. Nearby, in the same ancient ecosystem, appeared Miloconmingia fengjiawa, another early vertebrate candidate, even smaller and softer than Picaia itself. Neither animal projected dominance. Their bodies carried no mineralized defenses. They moved through an ocean already crowded with efficient predators and heavily armored competitors. Most likely, they occupied modest positions within the middle layers of the food web, feeding quietly while remaining vulnerable to almost everything around them. Anomalocaris could likely have consumed them effortlessly. That reality gives these early chordates a strange kind of weight. The entire vertebrate lineage, every future forest animal, every dinosaur crossing Mesozoic floodplains, every bird flying through modern skies, every whale moving through the deep ocean, and every human being looking back across this history, existed then only as a handful of soft-bodied swimmers, small enough to disappear into suspended sediment. Nothing about them guaranteed success. Their survival depended partly upon flexibility. The notochord-based body plan could adapt to changing environments and increasing body size without losing its fundamental organization. Segmental muscles allowed efficient movement through water. Concentrated sensory structures supported directional behavior in increasingly complex ecosystems. But in the Cambrian itself, these advantages remained potential rather than achievement. The lineage survived quietly, almost anonymously, through an ocean still intensifying around it. Predators improved their vision. Armor thickened across countless body forms. Burrowing strategies deepened. Feeding methods specialized. The pressure within the ecosystem continued rising from every direction, and these small chordates endured within that pressure without dominating it. For a long time, survival was enough. Long after the radiodonts vanished and the archaeocyath reefs dissolved from the oceans, trilobites continued walking across the seafloor. They survived environmental collapse, chemical upheaval, continental rearrangement, and extinction after extinction, persisting through spans of time so immense that entire mountain ranges rose and eroded away, while their descendants still moved beneath ancient seas. Among all Cambrian animals, none endured longer. Trilobites first appear in the fossil record approximately 521 million years ago, already widespread and anatomically sophisticated. They remain present until the end of the Permian extinction, roughly 252 million years ago. For around 270 million continuous years, they occupied nearly every ocean on Earth, becoming one of the most successful animal groups in the history of marine life. More than 20,000 species have been described from fossil deposits across the world. Some measured barely a millimeter in length, delicate enough to rest upon a fingertip. Others approached 72 centimeters, broad armored bodies, moving slowly across Paleozoic seafloors beneath dim marine light. Their anatomy followed a remarkably adaptable design. A central axial lobe ran down the middle of the body, flanked on each side by plural lobes that gave the group its name. At the front sat the cephalon, a hard shield containing the eyes, feeding structures, and sensory organs. Behind it extended a segmented thorax capable of controlled flexibility, followed by a fused tail structure called the pygidium. Beneath the armor moved rows of jointed legs used for walking, feeding, and interaction with sediment, while antennae projected forward, sensing chemical traces and subtle disturbances in surrounding water. This body plan proved versatile enough to occupy almost every ecological role available to medium-sized marine arthropods. Some trilobites hunted actively across the seafloor. Others filtered suspended particles from passing currents. Some scavenged decaying organic matter. Certain species spent much of their lives drifting within the water column itself. Entire ecosystems could contain multiple trilobite species operating simultaneously in different niches without directly competing with one another. Part of their resilience came from defense. Many trilobites possessed the ability to enroll, curling their segmented bodies inward until the vulnerable underside disappeared beneath hardened outer plates. The maneuver resembles the behavior of modern pillbugs, though perfected hundreds of millions of years earlier beneath Cambrian and Ordovician seas. Some fossilized specimens remain preserved in mid-enrollment, frozen in stone before the movement could fully close around them. Yet perhaps the most extraordinary feature of trilobites was their vision. Their compound eyes remain among the finest preserved visual systems in the entire fossil record. Unlike most animal eyes, trilobite lenses were constructed from calcite crystal, the same mineral found in limestone. Even after 500 million years, some lenses remain optically transparent enough for researchers to analyze their focal properties directly. Certain trilobite species evolved astonishingly sophisticated optical solutions. Some possessed aspheric calcite lenses capable of correcting spherical aberration, an issue that distorts images passing through curved surfaces. Others developed doublet lens systems composed of layered materials with different refractive properties, improving image clarity underwater. Human optical engineers independently discovered comparable solutions only within the last few centuries. Different trilobite groups also evolved different visual strategies. Some species possessed broad panoramic fields of view suited for detecting predators across open seafloors. Others developed highly specialized eyes adapted for dim, deeper water or bright, shallow environments. Through their fossils, paleontologists can reconstruct ancient marine conditions with remarkable precision. Particular trilobite species indicate specific oxygen levels, water depths, and temperature ranges, allowing entire vanished coastlines and continental shelves to be mapped through their distribution alone. In this way, trilobites became more than inhabitants of ancient oceans. They became record keepers of them. For hundreds of millions of years, they endured, while countless other Cambrian experiments disappeared entirely. Their bodies adapted repeatedly, without abandoning the underlying architecture that first appeared during the explosion of complex life. Oceans changed around them, climates shifted, ecosystems reorganized, yet trilobites persisted through nearly every transformation the Paleozoic world could impose. And still, even this extraordinary lineage would eventually meet an ocean no organism could fully survive unchanged. The Cambrian Ocean is often imagined as a continuous flowering of complexity, a long, uninterrupted expansion of life across ancient seas. Fossils seem to support this impression at first glance. New body plans appear, predators emerge, reefs rise from shallow shelves, and ecosystems spread through the water column with extraordinary speed. Yet hidden within the rock record are quieter interruptions, intervals where entire communities collapsed and portions of the experiment vanished almost as quickly as they arrived. The Cambrian Explosion was also a sequence of extinctions. One of the earliest major disruptions occurred around 517 million years ago. Across shallow marine environments, Archaeocyath reefs that had once spread across continental shelves declined abruptly. Their porous mineral structures disappeared from many ecosystems almost entirely. At the same time, large portions of the small shelly fauna vanished from the fossil record as well, reducing the extraordinary variety of early mineralized experiments that had characterized the beginning of the Cambrian. The leading explanation lies not in asteroid impacts or volcanic catastrophe, but in oxygen itself. During several intervals of the Cambrian, oxygen-depleted water appears to have expanded upward from deeper marine zones into shallower habitats where most complex ecosystems existed. These anoxia events transformed portions of the ocean into environments incapable of supporting animals adapted to oxygen-rich conditions. Communities that had evolved within newly ventilated shallow seas suddenly encountered suffocating water spreading silently across the continental shelves. The evidence survives chemically inside ancient carbonate rock. Researchers studying Cambrian sediments measure variations in carbon isotopes, particularly shifts in the ratio between carbon-13 and carbon-12 preserved within marine deposits. Under stable conditions, these ratios fluctuate within predictable limits. During ecosystem collapse, burial of organic material, and disruption of ocean chemistry alter the balance dramatically. The resulting excursions remain preserved in stone as faint geochemical signatures of biological crisis. Multiple such excursions occur throughout the Cambrian record. Among the most significant was the event now called the Steptoean positive carbon isotope excursion, often shortened to SPICE. Around 500 million years ago, carbon isotope values shifted sharply across marine sediments worldwide, signaling one of the largest chemical disturbances of the entire Cambrian period. Evidence suggests deep water anoxia intensified during this interval, reducing habitable marine zones and contributing to substantial extinction among trilobite populations. Even trilobites, with all their resilience and adaptability, did not pass unchanged through these disruptions. Cambrian trilobite communities repeatedly turned over across geological time. Entire families disappeared from marine ecosystems and were replaced by different groups adapted to altered environmental conditions. The trilobites surviving into the Ordovician world were anatomically and ecologically distinct from many of the forms dominating earlier Cambrian seas. Their lineage persisted, but not without continual restructuring beneath the pressure of extinction. And yet something deeper endured beneath these losses. The ecological framework established during the Cambrian remained remarkably stable even as species and groups vanished. Predators continued hunting prey. Filter feeders still processed suspended nutrients from passing currents. Burrowers churned sediment beneath the seafloor, while drifting organisms occupied the open water above. The architecture of the marine ecosystem survived repeated collapse events, even when the specific organisms inhabiting each role changed dramatically. In this sense, the Cambrian Ocean behaved less like a finished world and more like a system under constant revision. Extinction did not erase the larger structure. It reshaped who occupied it. By approximately 485 million years ago, another major extinction pulse marked the formal end of the Cambrian itself. Many groups strongly associated with Cambrian ecosystems declined or disappeared entirely, opening ecological space for the diversification that would follow during the Ordovician. Marine life expanded again afterward, but within boundaries already established during the earlier explosion of body plans. The ocean emerging from these extinctions was therefore both familiar and transformed. Much of the experimentation characteristic of the early Cambrian had been reduced, filtered, or lost. What remained became the foundation for future marine ecosystems across the Paleozoic world. Seen from a distance of 500 million years, the Cambrian no longer appears as a stable golden beginning. It resembles something far more dynamic, an unstable interval of invention repeatedly interrupted by collapse, where extinction and creation unfolded together inside the same ancient sea. If a human being could descend into the Cambrian Ocean without protection, the sea itself would feel unfamiliar long before any animal came into view. The chemistry of that water differed from the modern ocean in ways that shaped every organism moving within it. Its mineral balance, temperature structure, oxygen distribution, and dissolved gases created conditions unlike those of any sea alive today. The Cambrian Explosion did not emerge separately from this chemistry. It emerged because of it. One of the most important differences involved calcium. Cambrian seawater contained far higher concentrations of dissolved calcium ions than modern oceans. Mineral building material drifted through the water in extraordinary abundance, making it comparatively inexpensive for organisms to construct shells, spines, plates, and skeletal structures from calcium carbonate or calcium phosphate. Once evolution discovered the biological machinery necessary to extract and shape these minerals, the ocean itself effectively subsidized armor production. This abundance helps explain the sudden proliferation of hard parts across Cambrian ecosystems. Organisms did not evolve mineralized structures within a chemically neutral environment. They evolved them in a sea already saturated with the materials required to build them. The water was also warmer. Early Cambrian surface temperatures are estimated to have averaged roughly between 20 and 25 degrees Celsius globally, noticeably warmer than most modern oceans. Polar ice existed only minimally, if at all, and vast shallow shelf seas absorbed sunlight efficiently across submerged continental margins. In some tropical epicontinental seas, temperatures may have approached 30 degrees Celsius. Warm water accelerated biological activity, increased chemical reaction rates, and supported productive microbial and algal growth near the surface. Yet, despite this warmth and productivity, the Cambrian Ocean remained sharply divided vertically by oxygen. Surface waters grew increasingly oxygen-rich during this period, supporting larger and more active animals than earlier oceans could sustain. But deeper water often remained heavily depleted in oxygen, sometimes nearly devoid of it altogether. Between these zones existed a relatively shallow chemocline, a chemical boundary separating oxygenated upper water from anoxic depths below. Because this boundary sat closer to the surface than in most modern oceans, the habitable zone for complex life remained comparatively thin. Cambrian ecosystems concentrated heavily along shallow continental shelves where oxygenated water, sunlight, and nutrient availability overlapped most consistently. This concentration partly explains why nearly all major Cambrian fossil deposits preserve shallow marine communities rather than deep ocean ecosystems. Below the chemocline, conditions became hostile to most animal life. Bacterial communities inhabiting oxygen-poor water produced hydrogen sulfide through anaerobic metabolism. The gas accumulated in deeper marine layers and within sediment below the seafloor, creating chemically toxic environments incompatible with most complex organisms. Entire regions of the Cambrian Ocean likely remained biologically sparse because of this sulfur chemistry alone. In practical terms, the deep ocean functioned as a vertical barrier. Many Cambrian animals never crossed into it. Life crowded instead into relatively narrow bands of warm oxygenated water near the surface and along continental margins. Sea level amplified this concentration further. With almost no permanent polar ice locking water away on land, global sea levels stood significantly higher than today. Vast, low-lying continental regions lay submerged beneath shallow, productive seas. Much of the Cambrian world resembled an immense network of warm marine platforms extending across continental interiors, environments ideal for photosynthetic productivity and ecological diversification. At the same time, another long planetary transition was unfolding through the chemistry of iron itself. For much of earlier Earth history, dissolved iron circulated abundantly through oxygen-poor oceans. When oxygen encountered this iron, it precipitated into banded iron formations, alternating layers of iron oxide and silica accumulating across ancient seafloors. By the Cambrian, these formations were declining. Rising atmospheric and oceanic oxygen had permanently altered the iron cycle of the planet. The chemistry sustaining earlier oceans was fading away. All of these changes existed simultaneously within the same sea. Warm, mineral-rich surface water supported expanding ecosystems, while anoxic sulfur-bearing depths lingered below. Oxygenated shelves concentrated animal life into crowded, productive regions, while chemical boundaries restricted movement downward. The ocean smelled different, carried different, suspended minerals, and filtered light differently through its altered biological productivity. It was not simply an earlier version of the modern sea. It was another kind of ocean entirely, one whose chemistry shaped the appearance of everything living within it, from the color of the water to the bodies moving through it. Sunlight entering the Cambrian Ocean did not pass into the same kind of water that fills the modern sea. The color surrounding that light, the particles suspended within it, and the organisms absorbing and scattering it, produced a visual world unlike any ocean alive today. Reconstructing that world requires chemistry, optics, paleontology, and a certain degree of careful imagination. because the appearance of ancient water leaves no direct fossil behind. Near the surface, the sea was likely more green than blue. Modern open oceans often appear deep blue because they are comparatively poor in biological productivity. Clear water absorbs longer wavelengths of light while scattering shorter blue wavelengths back toward the surface. The Cambrian Ocean functioned differently. Dense concentrations of cyanobacteria and early algae filled shallow marine shelves with suspended organic material, tinting large regions of water green to blue, green in the same way productive coastal seas appear today. This upper layer shimmered with microscopic life. Sunlight diffused through drifting cells and suspended particles rather than penetrating cleanly downward through transparent water. The ocean surface would have carried a richer and softer coloration, less like the deep cobalt expanses of modern pelagic zones and more like vast illuminated shelves alive with suspended biology. Below that surface, the seafloor itself had changed appearance completely from earlier Precambrian environments. Before the Cambrian, the bottom of the sea was dominated visually by microbial mats, muted brown-gray surfaces stretching quietly across undisturbed sediment. By the Middle Cambrian, the substrate had become varied and textured. Pale archaeocyath reef structures rose upward in clustered formations. Sponge colonies introduced whites, creams, and orange tones into the environment. Burrow openings punctured darker sediment beneath drifting organic debris. Trilobites moved across the bottom, carrying calcite eyes that occasionally reflected fragments of filtered light back into the water. The seafloor had become visually complex because the ecosystem itself had become complex. Evidence from exceptionally preserved fossils suggests many Cambrian arthropods also possessed pigmentation patterns suited to this changing environment. Scientists studying preserved melanosomes, microscopic pigment-bearing organelles recovered from fossil tissue, have identified reddish-brown and black coloration in some Cambrian organisms. These colors likely functioned as camouflage against mineral-rich sediment, reef structures, and shadowed surfaces along the seafloor. But pigmentation represented only one response to vision. Another response was the complete abandonment of visible color altogether. As compound eyes spread through marine ecosystems, transparency became one of the most effective forms of concealment available to small drifting organisms. A transparent body blends directly into surrounding water without requiring armor, mineral investment, or active camouflage behavior. Many Cambrian zooplankton were almost certainly nearly invisible, except when illuminated from certain angles, just as countless gelatinous planktonic animals remain today. In a newly visual ocean, invisibility itself became an evolutionary strategy. The water also appears to have been more turbid than much of the modern open sea. Higher concentrations of suspended organic particles, algae, and microbial material scattered incoming sunlight more aggressively, reducing light penetration with depth. Even relatively shallow seafloors may have existed beneath dim, green, filtered illumination rather than clear, bright water. Beyond certain depths, darkness arrived quickly. This changing light environment shaped the perception of the predators moving through it. Anomalocaris, equipped with thousands of compound lenses in each eye, likely did not perceive the ocean in the detailed, continuous imagery familiar to human vision. Its sensory world was probably composed instead of shifting contrasts, movement patterns, brightness changes, and silhouettes suspended against varying light gradients. Yet, within the context of the Cambrian Ocean, this represented an extraordinary advance in perception. The sea had spent billions of years organized around chemistry and touch. Now it had become a world of visual detection. Movement could be seen. Shape could be recognized. Shadows carried meaning. The Cambrian Ocean therefore looked different at every level, from its green surface waters to the dim textured bottom below. Light filtered through richer biology, reflected from mineralized bodies, vanished into suspended sediment, and scattered across ecosystems crowded with organisms learning to hide, detect, mimic, and observe one another for the first time. And as these visual systems intensified, another sensory dimension quietly entered the ocean alongside them, one carried not by light, but by vibration, collision, and movement through water itself. Long before whales carried songs across entire ocean basins, before coral reefs crackled with shrimp and reef fish, before dolphins filled the sea with pulses of echolocation, the ocean existed in an almost complete state of biological silence. For billions of years, life drifted, absorbed nutrients, divided, and decayed without producing meaningful sound. Bacterial mats released chemicals into the water, but generated no acoustic world. The soft ediacaran organisms lying across ancient seafloors moved little and possessed no rigid structures capable of striking, scraping, or vibrating against one another. The Cambrian altered that silence gradually, then permanently. The first biological sounds in the ocean were not voices, they were consequences. Hard parts had entered the marine world, and once skeletons, shells, spines, and jointed limbs appeared, movement itself began producing noise. Trilobites, crossing mineral sediment, likely created faint scraping rhythms as articulated legs contacted limestone grains and shell fragments beneath them. Shell edges knocked gently against neighboring shells, where crowded animals competed for stable positions along the seafloor. Burrowing organisms displaced sediment in soft pulses and collapses beneath the substrate. Predation added sharper sounds to the water. The segmented appendages of radiodonts such as Anomalocaris closed around prey with physical force for the first time in Earth's history. Hard mouth plates, compressed, mineralized shells. Broken armor shifted across the seafloor after failed attacks or scavenging events. None of these sounds resembled the rich biological acoustics of modern oceans, yet together they marked a profound transition. Animal activity had become mechanically loud enough to disturb surrounding water. The ocean was beginning to acquire texture and sound as well as light. Some Cambrian arthropods may also have generated intentional vibrations through primitive forms of stridulation, the production of sound by rubbing body parts together. Modern arthropods perform this behavior constantly. Crickets scrape specialized structures to call across forests, lobsters vibrate body parts underwater, and countless insects communicate through friction-based sound production. Fossils cannot preserve these behaviors directly, but the anatomy required for mechanical contact between hardened structures almost certainly existed in some Cambrian species. Even more important than sound production was sound detection. Many Cambrian arthropods possessed sensory hairs known as setae distributed across limbs and body surfaces. These fine structures responded to subtle changes in water displacement, allowing animals to detect approaching movement through pressure waves traveling across the ocean. A swimming predator disturbed surrounding water before making physical contact. A struggling prey animal transmitted rhythmic pulses outward through the sea. Long before sophisticated hearing evolved, Cambrian organisms were already perceiving one another through hydrodynamic vibration. The ocean had become detectable through motion itself. Among early chordates, additional sensory systems may already have been emerging. Modern fish possess the lateral line, a distributed network of pressure-sensing organs capable of reading turbulence, direction, and nearby movement with extraordinary precision. Primitive functional ancestors of this system may have existed in Cambrian chordates, drifting weakly through shallow marine shelves. If so, even the earliest vertebrate relatives were navigating an ocean increasingly crowded with invisible signatures of movement and vibration. Yet, compared to modern seas, the Cambrian soundscape remained sparse. There were no whale migrations resonating across continents of water, no snapping shrimp generating underwater crackling loud enough to interfere with sonar, no fish choruses rising at dusk from coral reefs, no mammalian calls, no echolocation, no complex vocal communication. The sounds of the Cambrian were thin, accidental, and intermittent, small mechanical disturbances scattered across immense stretches of sea. And beneath the chemocline, below the oxygenated upper layers where most Cambrian animals lived, the ancient silence still remained intact. The deeper anoxic waters carried almost no biological movement at all. No scraping trilobites crossed the sediment there. No appendages struck prey. No sensory hairs detected approaching turbulence because there were almost no animals present to generate it. Hydrogen sulfide accumulated in darkness beneath the habitable zone, preserving a lower ocean that remained chemically hostile and nearly empty of complex life. Above that silent boundary, however, the living ocean was becoming increasingly perceptive. Vision had emerged, vibration sensing had spread. Organisms were processing movement, light, pressure, and spatial information in ways Earth had never seen before. And hidden inside these evolving animals, nervous systems were expanding to manage all of it. Somewhere in the shallow Cambrian seas, beneath drifting algae and suspended clouds of microscopic life, an arthropod was watching movement for the first time with something approaching true perception. Light struck thousands of compound lenses, pressure waves bent, sensory hairs along jointed limbs. Chemical traces drifted through seawater toward delicate antennae. Muscles adjusted position continuously as the animal crossed uneven sediment. None of these signals meant anything unless they could be gathered, compared, prioritized, and transformed into action. The Cambrian Ocean had created a new problem for life. Sensing the environment was no longer enough. Animals now needed to process the environment. For billions of years, simple nerve nets and localized reflexes had been sufficient. The Ediacaran world demanded little computation because almost nothing moved rapidly and almost nothing hunted. But once eyes evolved and predators began selecting prey visually, reaction time became a matter of survival. Organisms capable of interpreting information faster gained immediate evolutionary advantage. The ocean was no longer a passive chemical landscape. It had become a constantly changing field of motion, threat, opportunity, and escape. Under that pressure, nervous systems expanded. One of the clearest pieces of evidence comes from Fuxianhuaya protensa, a primitive arthropod preserved in the Chengjiang deposits approximately 520 million years ago. Within several extraordinary fossils, researchers identified preserved traces of neural tissue, including a recognizable brain structure and paired optic lobes. These optic regions processed visual input separately from other sensory signals, revealing something profound. The relationship between vision and brain complexity is visible directly in the fossil record itself. The moment eyes appeared, brains began reorganizing around them. Fuxianhuia possessed a nervous system already divided into functional regions. Sensory information did not flow through the body randomly. Different structures performed different tasks. Signals from the eyes moved toward optic processing centers. Other nerve pathways controlled limbs and body movement. Neural specialization had already emerged surprisingly early in animal history. Across Cambrian arthropods, Nervous systems also displayed another important feature: segmental organization. The body itself was divided into repeated sections, and the nervous system mirrored that structure. Chains of nerve clusters extended beneath the animal, with different ganglia coordinating different body segments semi-independently. A trilobite walking across the seafloor did not require a single centralized command for every limb movement. Local neural circuits controlled much of the motion automatically, while higher processing regions integrated broader sensory information. That architecture survives almost unchanged in modern arthropods and segmented worms today. At the same time, sensory demands were multiplying rapidly. A Cambrian arthropod equipped with compound eyes, mechanoreceptive setae, chemical sensing antennae, and proprioceptive awareness of limb position needed to merge all these streams of information continuously. The animal had to distinguish prey from sediment disturbance, current movement from approaching danger, and background noise from meaningful vibration. The Ediacaran world had never required anything remotely comparable. Predators accelerated the pressure further. As hunting strategies improved, prey species capable of reacting more quickly gained advantage. Faster escape responses spread through populations. Better directional awareness improved survival. Nervous systems capable of coordinating rapid movement, accurate orientation, and flexible behavior became increasingly valuable. Predator and prey were now shaping one another neurologically as much as physically. The arms race had entered cognition. Among early chordates drifting through the same seas, another neural architecture was taking shape. Instead of the ventral nerve arrangement common in arthropods, chordates carried a dorsal nerve cord running along the back above the notochord. In fossils more than 520 million years old, faint preserved traces reveal this elongated structure extending through the body. That narrow cord is the precursor to every vertebrate, spinal cord, and brain that would ever exist. Inside those small, soft-bodied chordates already lived the foundational geometry of fish, reptiles, birds, mammals, and eventually human beings. The infrastructure of vertebrate thought began not in dominance, but in vulnerability. Hidden among armored predators and crowded seafloors during a brief interval, when evolution was experimenting faster than at any other known point in Earth's history. The ocean now possessed vision, locomotion, armor, predation, and nervous systems capable of integrating them together into behavior. Separate innovations were beginning to interlock into something larger, a living structure of relationships, pressures, dependencies, and energy flow spreading through the Cambrian Sea. In the shallow Cambrian seas, life was no longer scattered into isolated experiments, drifting through empty water. By roughly 510 million years ago, the ocean had arranged itself into layered relationships of energy, movement, feeding, death, and renewal. Different organisms depended on one another directly or indirectly, not as occasional interactions but as permanent structure. The sea had become an ecosystem in the modern sense, organized vertically from the illuminated surface to the sediment below. Near the top of the water column, beneath sunlight filtering through green-tinted water rich with suspended microorganisms, cyanobacteria and early algae formed the productive foundation of everything above them. Through photosynthesis, they converted sunlight and dissolved carbon dioxide into organic matter, building living tissue from light itself. Countless microscopic cells drifted through the photic zone, reproducing continuously in warm, shallow seas spread across flooded continental shelves. From that suspended productivity emerged the first true consumers. Zooplankton grazed on phytoplankton in enormous numbers, packaging microscopic life into animal tissue that larger organisms could eat. Small drifting larvae belonging to bottom-dwelling species passed through the water column, feeding before settling onto the seafloor later in life. Sponges filtered seawater constantly through porous bodies, removing organic particles one flow at a time. Brachiopods opened mineral shells slightly against the current and extracted suspended nutrients with delicate feeding structures hidden inside. Across the seafloor, small grazers moved between burrows, reef structures, and sediment surfaces. Organisms such as Wiwaxia scraped microbial films and organic debris from hard surfaces, while primitive mollusks searched slowly across encrusted substrate. Deposit feeders processed sediment directly, extracting nutrition from decaying material buried beneath the surface. Nothing was wasted for long. Dead tissue, discarded waste, and sinking organic particles entered a continuous cycle of consumption and recycling. Above them moved the secondary consumers. Predatory priapulid worms pushed through sediment, searching for smaller animals hidden beneath the seafloor. Early crustacean-like arthropods hunted drifting prey in the water column or scavenged organic remains along reef structures. Juvenile radiodonts occupied intermediate predatory roles before growing large enough to challenge bigger prey. Each ecological level depended on the stability of the ones beneath it. At the top of the system cruised the apex predators. Large radiodonts, such as Anomalocaris, moved through open water using sophisticated eyes and flexible swimming lobes to locate prey visually. Their existence represented something entirely new in Earth's history: predators large enough and neurologically advanced enough to shape the behavior of entire communities around them. Prey species altered movement patterns, body structures, feeding strategies, and habitat preferences under the constant pressure of being hunted. Yet the ecosystem's stability did not depend on any single organism. One of the most important developments already visible in Cambrian communities was ecological redundancy. Multiple species occupied overlapping roles simultaneously. Several types of filter feeders processed suspended particles. Different scavengers recycled organic matter along the seafloor. Numerous predators targeted similar prey using different strategies. If one lineage declined or disappeared, another could continue performing much of the same ecological function. That redundancy gave the system resilience. At the same time, species increasingly divided the environment among themselves through habitat partitioning. Some organisms specialized in soft sediment, others in reef surfaces, others in open water. Certain animals occupied shallower illuminated zones, while others remained deeper near the boundary of low oxygen water. Some fed during constant movement, others while anchored permanently to the substrate. The Cambrian Ocean was no longer dominated by generalists surviving wherever conditions allowed. It was being partitioned into ecological territories occupied by specialists. Analyses of Burgess Shale communities reveal something especially striking about this arrangement. When researchers examine predator-prey ratios, species abundance patterns, and niche overlap distributions within these Cambrian ecosystems, the statistical relationships resemble those found in modern marine ecosystems remarkably closely. The system was not primitive in organizational logic. Within only a few tens of millions of years, the ocean had assembled a structurally mature food web with recognizable ecological architecture. The framework of the modern living world already existed. Energy flowed downward from sunlight into plankton, upward through predators, and back again through decomposers, processing death into nutrients. Different species occupied distinct roles within interconnected networks, stable enough to persist through environmental fluctuation, competition, and extinction. The ecosystem was complete now, but completion did not mean permanence. Beneath its apparent stability, pressure was already building inside the chemistry and climate of the Cambrian Ocean itself. Across the Cambrian seafloor, among trilobite tracks, sponge colonies, and drifting clouds of suspended sediment, moved a small, armored animal whose identity remains unsettled more than 500 million years later. Its fossils appear so frequently in Burgess Shale deposits that paleontologists know its body in remarkable detail, yet that detail has not resolved the deeper problem. Wiwaxia corrugata sits in paleontology like a sentence written in a language only partially translated, familiar in fragments, uncertain in meaning. At first glance, Wiwaxia appears almost defensive to excess. Its body was oval-shaped and low against the seafloor, ranging from roughly 1.5 to 5 centimeters in length. Across its upper surface lay dense overlapping scales arranged like layered armor plates, while 2 rows of long blade-shaped spines projected upward and outward from the back. In life, those spines likely swayed slightly with movement and current, transforming the animal into a moving forest of mineralized points. The arrangement was not elegant in the streamlined sense of later marine animals. It looked assembled for endurance rather than speed. Yet beneath all that protection, the underside remained entirely soft. Wiwaxia moved directly across the substrate on a muscular ventral foot, similar to the creeping locomotion used by modern slugs and snails. Its exposed underside pressed continuously against the sediment as it traveled slowly over the seafloor surface. The contrast was striking: extreme armor above, complete vulnerability below. If overturned or lifted from the substrate, the animal would have lost the protection its body depended on. Its feeding apparatus complicates the picture further. Inside the mouth was a radula, a ribbon-like structure lined with rows of tiny teeth used to scrape organic material from surfaces. Modern mollusks use the same mechanism. Snails rasp algae from rock using radulae. Chitons graze microbial films the same way. Even cephalopods possess modified radular structures deeper within their feeding anatomy. The presence of a radula in Wiwaxia strongly suggests some relationship to mollusks, and for many researchers, this connection remains the most convincing interpretation. But the rest of the body resists that conclusion. Certain details of Wiwaxia's anatomy align poorly with known mollusks. The arrangement of its sclerites, the geometry of its body, and aspects of its preserved anatomy resemble polychaete annelid worms more closely in some analyses. Other researchers argue that neither interpretation fits comfortably because Wiwaxia may belong to a lineage positioned outside both groups entirely, representing a separate evolutionary branch that later disappeared. The disagreement persists because the Cambrian Ocean was still experimenting with combinations of anatomical features that would later become separated into distinct animal lineages. Traits modern biology treats as defining characteristics of specific groups were not yet distributed so cleanly. Evolutionary boundaries remained fluid. Wiwaxia exists directly inside that uncertainty. Its importance, therefore, extends beyond the animal itself. If it truly belongs to the mollusk lineage, then the ancestry of mollusks stretches deep into the Cambrian in armored forms connected to the small shelly fauna. The scattered sclerites preserved globally in Cambrian sediments may partly represent the disassembled remains of Wiwaxia, like animals moving across ancient seafloors worldwide. If it instead belongs to annelids or to an entirely extinct lineage, then those same sclerites become evidence of another evolutionary experiment that flourished briefly before vanishing without descendants. Either way, Wiwaxia was extraordinarily successful in its own time. Its isolated scales and spines occur throughout Cambrian rock layers across multiple continents because the armored elements separated easily after death and fossilized independently. Even when the animal itself disappeared into decay, fragments of its protective covering remained scattered across the seafloor, eventually buried into stone. That abundance gives paleontologists an unusual intimacy with an organism they still cannot classify with confidence. The uncertainty surrounding Wiwaxia is not a sign that science has failed to understand the Cambrian. It is evidence that the Cambrian genuinely occupied a different evolutionary condition from the modern world. The animal groups familiar today had not yet settled into fixed anatomical identities. Features that later became exclusive were still mingling freely through bodies shaped by a rapidly changing ocean. Movement had existed before the Cambrian in limited forms. Slow contractions across microbial surfaces, drifting bodies responding passively to current, slight repositioning through sediment. But by the middle of the Cambrian, the ocean had become a place of active trajectories. Animals now crossed distances intentionally. They pursued prey, escaped danger, explored new habitats, and occupied separate physical layers of the sea through fundamentally different methods of locomotion. The water itself had become structured by motion. One of the most important innovations behind this transformation was the arthropod body plan. Jointed appendages connected to a rigid external skeleton allowed a precision of movement Earth had never seen before. The exoskeleton provided stable attachment points for muscles, while articulated joints permitted controlled bending in specific directions. With this arrangement, limbs could specialize. Some evolved for walking, others for swimming, grasping, burrowing, or feeding. This process, sometimes called arthropodization, changed the mechanical possibilities available to life. Across the Cambrian seafloor, trilobites moved over sediment on coordinated rows of jointed legs beneath hard segmented bodies. Their movement was not smooth in the vertebrate sense, but rhythmically precise, each limb operating in carefully timed sequence against the substrate. Trace fossils preserved in Cambrian rock still record these pathways as faint parallel impressions crossing ancient marine sediment. Other arthropods adapted the same mechanical principles to open water. Large radiodonts, such as Anomalocaris, swam through lateral lobe undulation. Flexible fins lining the sides of the body beat in coordinated waves from front to back, generating smooth propulsion through the water column. This movement style combined efficiency with maneuverability, allowing large predators to maintain sustained motion while adjusting direction rapidly around prey or obstacles. The body itself remained relatively stable, while propulsion traveled along the sides like moving ripples through fabric. In early chordates, a completely different locomotor architecture emerged. Instead of jointed appendages or lateral fins, small, soft-bodied swimmers relied on a notochord running the length of the body as an internal stabilizing structure. Segmented muscles contracted alternately along either side, producing traveling sinusoidal waves that pushed against surrounding water. The animal advanced through coordinated bending rather than limb movement. Every modern fish still swims through variations of this same principle. The essential mechanics of vertebrate locomotion were already established in Cambrian seas more than 500 million years ago. Other lineages discovered entirely different solutions. By approximately 500 million years ago, early cephalopods had evolved jet propulsion. Water drawn into the body cavity could be expelled rapidly through a funnel-like structure, producing sudden directional movement. This was the earliest known appearance of jet propulsion in animal history. Instead of relying on limbs or undulating bodies, these organisms transformed water pressure itself into locomotion. Beneath the sediment surface, priapulid worms used yet another strategy. Their movement depended on hydraulic pressure generated inside fluid-filled body cavities. Circular muscles contracted to force portions of the body forward into soft sediment, after which longitudinal muscles pulled the remainder of the animal behind. Modern priapulids still employ almost exactly the same method today. In some parts of the marine world, Cambrian mechanics continue almost unchanged beneath the seafloor. Not every organism relied on active propulsion. Many smaller Cambrian animals adopted passive movement strategies instead. Elaborate spines increased surface area and slowed sinking through water. Flattened body shapes generated drag that allowed organisms to hover within productive layers of the ocean with minimal energy expenditure. Others approached neutral buoyancy through body composition and fluid balance, remaining suspended in the water column almost effortlessly while currents transported them gradually across marine shelves. Each locomotion strategy opened access to different ecological space. Walking species exploited hard surfaces and sediment. Burrowers occupied the subsurface environment inaccessible to swimmers. Passive drifters remained in nutrient-rich upper waters. Fast predators crossed between layers searching for prey. Locomotion diversified the ocean physically, reducing direct competition by allowing organisms to partition the environment according to how they moved through it. The Cambrian Sea had become a world of intersecting trajectories, limbs scraping sediment below, drifting plankton suspended above, predators undulating through open water, buried worms forcing themselves hydraulically through mud beneath them all. And every movement required energy. Every muscle contraction, every swimming wave, every grasping appendage depended on the same underlying question that now shaped the entire ocean: how animals found food within this expanding world of motion. Long before forests existed on land, before birds crossed the sky or mammals walked across continents, the Cambrian Ocean had already become a place of relentless experimentation in how energy could be captured from the environment. Every surface, every current, every layer of sediment and water represented an opportunity. And because animal body plans were diverging so rapidly, feeding itself began fragmenting into dozens of separate mechanical solutions almost simultaneously. Some strategies depended on stillness. Sponges, brachiopods, and archaeocyaths anchored themselves to the seafloor and transformed water movement into nutrition. Tiny, suspended particles drifted constantly through Cambrian seas: bacteria, algae, organic debris, microscopic plankton. By pulling seawater through pores, chambers, or ciliated feeding structures, these animals filtered food directly from the current. The ocean itself became part of their digestive system, delivering nutrition continuously without pursuit or struggle. It was among the lowest energy feeding strategies available to animal life, and it scaled efficiently across dense reef communities where thousands of organisms filtered the same moving water simultaneously. Other animals fed not from the water above them, but from the sediment below. Deposit feeders crossed the seafloor methodically, consuming the uppermost layer of mud and extracting organic material trapped within it. Their movements left dense networks of feeding traces preserved in Cambrian rock. In some fossil beds, these overlapping trails almost completely cover the ancient sediment surface, evidence of animals systematically processing enormous areas of seafloor for nutrition. The bacterial mat worlds of earlier ages had become edible terrain. Grazers evolved a more precise relationship with surfaces. Wiwaxia and early mollusks carried radulae, flexible ribbons lined with microscopic teeth capable of scraping thin organic films from rock, shell, and microbial crust. With controlled muscular motion, the radula functioned like a biological rasp, removing algae and bacteria layer by layer. Modern snails still feed through essentially the same mechanism today. Across more than 500 million years, the engineering proved so effective that evolution had little reason to replace it. Elsewhere in the water column, feeding became active pursuit. Large radiodonts such as Anomalocaris no longer waited for nutrition to arrive. They searched for it visually. Compound eyes scanned movement through suspended green water, while segmented frontal appendages closed around prey with coordinated precision. Prey could flee, maneuver, hide, or defend itself, which meant predators required increasingly sophisticated sensory systems and locomotion to succeed. Feeding had become an interaction between nervous systems rather than a passive relationship with current or sediment. Not every predator relied on speed. Priapulid worms embedded themselves beneath the sediment with only toothed mouths exposed at the surface. Hidden almost entirely from view, they waited motionless until another organism passed within reach. Then the mouth extended outward through hydraulic pressure, armed with arrays of inward-pointing teeth capable of seizing soft prey rapidly before retreating underground again. More than 30 Cambrian priapulid species are known, many displaying remarkably intricate feeding structures arranged in concentric rings and hardened grasping elements. Scavenging emerged alongside predation. Once animals grew larger and populations denser, the ocean began producing substantial carcasses for the first time. Dead trilobites, discarded molts, injured swimmers, partially consumed prey, all became concentrated packets of organic matter descending through the ecosystem. Mobile arthropods and sediment feeders processed this material quickly, recycling nutrients back into the food web. Death itself had become a new ecological resource. Parasitism may also have existed, though the fossil record preserves only uncertain hints. Specialized parasitic anatomies require close evolutionary relationships between host and parasite, and the Cambrian Explosion may still have been too young for many of these intricate dependencies to fully emerge. The ocean was crowded with experimentation, but some forms of biological intimacy likely had not yet stabilized. Again and again, unrelated lineages converged on similar answers. Filter feeding evolved independently in multiple groups. Spines appeared across distant body plans. Radula, like scraping structures, emerged more than once. The same environmental pressures repeatedly produced similar mechanical solutions, as though the Cambrian Ocean contained a limited set of workable strategies and evolution kept rediscovering them from different directions. Beneath all this complexity, however, lay something older and slower than biology itself. Continental movement, ocean chemistry, volcanic cycles, atmospheric change, the geological systems shaping the planet had created the conditions that allowed this extraordinary flowering of animal life. And those same systems had already begun changing again. At the boundary between the Cambrian and the Ordovician, the ocean did not gently evolve into something richer and more stable. It fractured, reorganized, and shed many of the forms that had once defined it. The great experimental surge that had filled the seas with impossible anatomies and unfamiliar architectures began narrowing into something more selective. Entire groups declined or vanished. Trilobite diversity contracted sharply. Many components of the small shelly fauna disappeared from the fossil record. Cambrian radiolarian communities shifted or collapsed. The strange balance that had briefly allowed so many body plans to coexist was breaking apart. Yet the ecological spaces left behind did not remain empty for long. Surviving lineages expanded into them almost immediately. Organisms descended from Cambrian ancestors diversified into new forms, occupying old feeding strategies, reef structures, predator roles, and sediment niches with modified anatomy and increasingly specialized behavior. The ocean was no longer inventing fundamentally new kinds of animals at the same pace. Instead, it was refining the survivors. Around 480 million years ago, the Great Ordovician biodiversification event accelerated across marine ecosystems. Species counts rose dramatically. Reef systems became larger and more structurally complex. Food webs thickened with increasing specialization. Suspension feeders diversified across continental shelves, while swimming predators occupied more levels of the water column simultaneously. But beneath all this expansion, the underlying architecture remained Cambrian. No new major animal phyla appeared. The great frameworks had already been established during that earlier interval when the ocean first became populated by vision, armor, locomotion, and predation. Every later marine ecosystem, no matter how different in detail, would operate within boundaries defined during the Cambrian transformation. The arthropod body plan endured, its segmented exoskeleton and jointed appendages radiating outward into crustaceans, insects, spiders, and countless marine descendants. Today, arthropods account for the overwhelming majority of known animal species on Earth. The chordate blueprint survived as well, preserving the dorsal nerve cord and supportive internal axis that would eventually produce fish, amphibians, reptiles, birds, mammals, and human beings. Mollusks continued through snails, clams, squid, and octopuses. Echinoderms persisted as starfish, sea urchins, and sea cucumbers. Annelids remained within marine worms and terrestrial earthworms. Sponges endured almost unchanged. Cnidarians carried forward into jellyfish, coral, and sea anemones. Nearly every complex animal alive today belongs to lineages already present in Cambrian seas. The ocean that generated these body plans, however, no longer exists. Its chemistry has been rewritten by 500 million years of biological feedback. Calcium concentrations shifted. Atmospheric oxygen rose further. Deep ocean circulation reorganized repeatedly as continents drifted across the globe. The shallow epicontinental seas that once flooded continental interiors disappeared and returned in different forms through later geological ages. The green, mineral-rich Cambrian waters became other oceans entirely. Even the continents themselves have forgotten the arrangement they held then. Tropical seafloors became mountain ranges through tectonic collision. Ancient reefs rose into cold alpine air. Sediments once buried beneath shallow equatorial water now rest thousands of meters above sea level, carrying fossil impressions of trilobites and radiodonts into landscapes of snow and stone. But the organizational logic created during the Cambrian never vanished. Primary producers capturing sunlight near the surface, consumers feeding on drifting life and sedimented remains, predators tracking motion through the water column, decomposers returning nutrients to circulation. Vertical layering, ecological redundancy, habitat partitioning, complex interdependence between species occupying different depths and niches. Every modern ocean still follows this arrangement. The details changed. The architecture remained, and somewhere within that architecture, carried continuously through extinction after extinction, through continents splitting and oceans closing, through every catastrophe that followed, The small vulnerable chordates of the Cambrian continued surviving long enough to become creatures capable of looking backward across 500 million years and recognizing the outline of themselves in the first ocean that ever learned how to live this way. In certain corners of the modern ocean, and even in damp forests on land, fragments of the Cambrian world still persist. They do not survive as frozen relics untouched by time, because evolution never truly stops. Genes shift, populations divide, environments impose subtle pressure generation after generation. Yet some lineages change so slowly in their underlying architecture that their bodies still echo organisms that crawled or burrowed through Cambrian sediment more than 500 million years ago. The phrase living fossil is often used for such animals, though it can be misleading. These organisms are not ancient in the sense of being unchanged individuals carried through deep time. They are modern species. What endured was the blueprint itself, a body plan so mechanically successful that wholesale redesign never became necessary. Among the clearest examples are the priapulid worms. In Cambrian seas, priapulids occupied the role of ambush predator beneath the sediment surface. Their bodies operated through hydraulic pressure, muscles compressing fluid-filled internal cavities to force the worm forward through mud. Ringed arrays of hardened teeth surrounded extendable mouths capable of seizing prey rapidly before retreating underground again. Modern priapulids still hunt this way. Roughly 20 living species remain today, mostly in cold marine sediment, using the same burrowing mechanics and the same anatomical logic their Cambrian ancestors used more than 520 million years ago. The resemblance is so close that if one were transported backward into Cambrian water, it would not appear radically out of place. Velvet worms carry another surviving thread from that vanished ocean. Their ancestors belonged to the lobopodians, soft-bodied animals with stubby paired limbs moving slowly across ancient seafloors. Hallucigenia represented one strange branch of this broader group, but the fundamental limb structure survived through descendants that eventually left marine environments and entered terrestrial ecosystems during the Paleozoic. Modern velvet worms still walk on soft lobopod legs operated through internal hydraulic pressure rather than rigid skeletons. Their movement retains something distinctly ancient, as though the mechanics of the Cambrian seafloor were still faintly visible beneath leaf litter and moss. Linguid brachiopods preserve another remarkably conservative design. Anchored to sediment by fleshy stalks, these filter feeders draw water through paired shells and extract suspended particles from ocean currents. Fossils from Cambrian rock are astonishingly similar to some living forms. Certain genera persisted with relatively modest structural change across nearly 500 million years of shifting oceans, extinctions, and continental rearrangements. The shell shape, feeding mechanism, and general anatomy proved durable enough to survive events that erased countless other lineages entirely. Even horseshoe crabs, though younger in their recognizable form, carry deep roots extending toward Cambrian arthropod ancestry. They are not true crabs, but chelicerates, relatives of spiders and scorpions. Their armored bodies, broad protective carapaces, compound eyes, and spined walking limbs preserve a mechanical style that has remained broadly stable for more than 450 million years. Modern coastlines still receive these ancient-shaped animals during spawning migrations, their bodies moving through shallow water with a geometry older than forests, dinosaurs, or flowering plants. What these conservative lineages reveal is not that evolution aims toward perfection. If that were true, the Cambrian Ocean would not have produced so many anatomies that disappeared forever. Success in evolutionary history is not a judgment on elegance or complexity. Many extinct Cambrian organisms were highly specialized, mechanically sophisticated, and ecologically effective within the environments they inhabited. Their disappearance does not mean they were inferior designs. Survival depended on circumstance. A cooling ocean, a changing chemistry, a shifting coastline, an extinction pulse moving through food webs. These forces determined which lineages remained positioned inside environments they could still tolerate. Some organisms happened to occupy ecological roles resilient to disruption. Others did not. The difference between persistence and disappearance was often astonishingly narrow. The ocean carried no preference. It preserved some body plans across 500 million years while erasing others almost completely. Not because one deserved endurance more than another, but because time, geology, chemistry, and chance moved together through Earth's history without intention, allowing certain fragile threads to continue while countless others dissolve silently into stone. Even after more than a century of excavation, imaging, chemical analysis, and argument, the Cambrian explosion remains partially obscured behind gaps that science has not yet managed to cross. The outlines are visible. The broad sequence is understood. But many of the deepest questions surrounding the event still resist closure, as though the ocean that produced these animals continues withholding part of its history beneath layers of missing sediment and vanished coastline. One uncertainty concerns tempo. How quickly did the Cambrian explosion truly occur? Early estimates stretched diversification across tens of millions of years, giving evolution long intervals to experiment gradually with anatomy and ecology. But as radiometric dating techniques improved, the timeline began compressing. Some analyses now suggest that the main pulse of diversification may have unfolded within as little as 5 to 10 million years. On geological timescales, this is extraordinarily rapid. Entire body plans appear, diversify, and establish stable ecosystems within a window so narrow that it challenges traditional assumptions about the pace at which natural selection alone can generate profound anatomical innovation. Another unresolved problem reaches further backward into time. Molecular clock studies, which estimate lineage divergence through accumulated genetic mutations, consistently imply that major animal groups separated long before their sudden appearance in Cambrian rock. Some calculations place the origins of animal phyla as far back as 800 million years ago, deep within the Precambrian world where the fossil record remains largely silent. This creates a tension between genetics and paleontology that has never been fully resolved. Perhaps early animals were so soft-bodied, microscopic, or geographically restricted that they simply failed to fossilize. Perhaps entire ecosystems existed in environments unfavorable to preservation. Or perhaps the molecular clocks themselves require recalibration, their assumptions about mutation rates drifting subtly across immense spans of time. The disagreement persists because both lines of evidence contain persuasive strengths and unavoidable uncertainties. There is also the problem of absence itself. Every extraordinary Cambrian fossil deposit represents only a tiny fragment of a vast planet. The Burgess Shale records one ancient marine slope community. Xinjiang captures another ecosystem in a different region and interval. Sirius Passet, Xinjiang, and other deposits illuminate additional isolated windows. But the overwhelming majority of the Cambrian Ocean remains invisible. The open pelagic waters, the deepest basins below the chemocline, polar regions, tropical offshore systems, Entire continental margins may have hosted communities unlike anything preserved in known deposits. Those organisms are gone without record. The fossil evidence that survives is therefore not a complete archive but a scattered collection of rare moments where geology interrupted decay quickly enough to preserve soft tissue before it vanished. Between those moments stretches enormous silence. That silence continues, shrinking slowly as new discoveries emerge. In 2019, researchers formally described the Xinjiang biota from Hubei province in China, a deposit approximately 518 million years old, preserving extraordinary soft-bodied detail. Despite overlapping in age with Xinjiang and approaching the older Cambrian communities already known, Xinjiang revealed a notably different species composition. Organisms absent from one deposit appeared abundant in another. Entire ecological structures shifted from region to region. New species continue to be described from these rocks, altering interpretations almost year by year. And still, the central question remains unsettled. Was the Cambrian explosion driven primarily by environmental change, rising oxygen, altered ocean chemistry, warming shallow seas? Was it ecological, a cascading arms race between predators and prey once vision and mobility emerged? Was it genetic, involving developmental innovations that suddenly allowed animal bodies to diversify into radically new forms? Or did the event require all these conditions simultaneously, interacting in ways too complex for a single explanatory model? No consensus exists. The Cambrian Ocean is not a solved chapter resting quietly in textbooks. It remains an active research frontier shaped continuously by new fossils, new dating methods, molecular biology, synchrotron imaging, geochemical analysis, and reinterpretations of specimens collected generations ago. Every few years, the picture shifts again, revealing that the first complex ocean on Earth still contains regions of darkness where understanding has not yet fully reached. Among the countless anatomies drifting, burrowing, swimming, and hunting through Cambrian water, the lineage that would eventually produce human beings was almost invisible. It carried no mineral armor. It possessed no crushing appendages, no spined exoskeleton, no elaborate defensive covering. In an ocean dominated by arthropods with hardened shells and predators already optimized for visual hunting, the earliest chordates occupied a precarious position close to the lower levels of the food web. They were small enough to disappear inside another animal's mouth without resistance. Picaia, Hycowithus, Miloconmingia, these soft-bodied swimmers measured only a few centimeters in length, their bodies flexed through alternating muscle contractions arranged around a supportive notochord. They sensed the world through primitive sensory structures and survived by remaining mobile in a crowded ecosystem increasingly shaped by predation. Nothing about them suggested future dominance. Nothing in their anatomy announced mammals, primates, language, or thought. They were simply one lineage among many, and their continuation was never guaranteed. During the Cambrian, repeated oxygen depletion events spread through marine environments as shifting ocean chemistry periodically pushed anoxic water upward into shallow shelf ecosystems. If any of these events had expanded slightly further across continental margins, the environment supporting early chordates could have collapsed entirely. Their populations were small, localized, and ecologically vulnerable. Extinction would not have required catastrophe on a planetary scale. A subtle chemical shift extending across the wrong coastline may have been enough. Predation represented another narrow threshold. Anomalocaris and other visual hunters already possessed sophisticated eyes, rapid swimming mechanics, and grasping appendages capable of processing soft prey efficiently. If these predators had specialized more aggressively towards small chordates, or if early vertebrate ancestors had occupied slightly more exposed ecological positions, the lineage might have been reduced faster than it could diversify. The Cambrian Ocean contained many anatomically promising groups that vanished permanently despite millions of years of persistence. Survival did not belong automatically to potential. Stephen Jay Gould later framed this uncertainty through a thought experiment that became central to discussions of contingency in evolution. If Earth's history could somehow be replayed from the Cambrian onward, with only minor changes in initial conditions, a slightly warmer ocean, a different continental arrangement, a more severe extinction interval, the resulting world would likely diverge profoundly from the one now existing. Vertebrates might never emerge as the dominant, large-bodied lineage. Arthropods, mollusks, or entirely different descendants of Cambrian experimentation could occupy ecological roles, later inherited by mammals. The future was not predetermined inside the Cambrian Ocean. What ultimately allowed the chordate lineage to endure was not obvious superiority, but unusual flexibility. The notochord-based body architecture proved remarkably adaptable across changing environments and body scales. Sensory structures could become more elaborate. Muscular systems could support larger bodies. Internal skeletons could strengthen while preserving mobility. The same fundamental design could move through shallow marine shelves, freshwater rivers, coastal swamps, forests, deserts, and eventually open air. But in the Cambrian itself, none of this had happened yet. The lineage existed only as possibility. Those fragile swimmers, drifting through green mineral-rich water, carried within them anatomical capacities that would not fully unfold for hundreds of millions of years. Dinosaurs, whales, bats, elephants, and human beings were not visible in any direct sense. The blueprint contained room for expansion, but not certainty of outcome. Every person alive today descends from organisms that survived this unstable interval through combinations of adaptability, geography, timing, and chance. The continuity stretches backward through every extinction boundary, every continental collision, every climate shift, toward animals so small and vulnerable that their disappearance would scarcely have altered the Cambrian ecosystem around them. Yet they persisted, and because they persisted, nervous systems continued increasing in complexity. Eyes refined themselves further. Memory, prediction, communication, abstraction, and reflection gradually emerged from biological architectures first assembled in those ancient seas. The Cambrian Ocean, therefore, represents more than the origin of modern ecosystems or the diversification of animal anatomy. It marks the interval in Earth's history when the physical conditions necessary for awareness began organizing themselves into durable form, allowing matter not only to live and reproduce, but eventually to observe its own origins and remember the ocean where that possibility first survived. Long before forests spread across continents or vertebrates walked onto land, the ocean had already become a machine for generating possibility. Its currents carried dissolved minerals through shallow continental seas. Its chemistry shifted gradually through volcanic outgassing, weathering rock, and microbial activity. Oxygen accumulated unevenly through the water column, while calcium and phosphorus circulated through expanding ecosystems. None of these processes possessed intention. They were simply physical conditions interacting across immense spans of time. Yet, from those interactions emerged the Cambrian world. The ocean did not move toward complexity as though following a destination. It established a set of thresholds, and once those thresholds were crossed, life responded with extraordinary speed. Oxygen concentrations rose high enough to support metabolically expensive movement. Calcium became abundant enough that shells and armor could be constructed with relatively little biological cost. Shallow continental shelves expanded beneath warm seas, concentrating nutrients and organisms into crowded ecological spaces where competition intensified continuously. The result was not planned. It was produced. Some researchers argue that once oxygen passed a certain metabolic boundary, complex animal life became effectively unavoidable. Chemistry itself may have constrained the possibilities strongly enough that multicellular organisms with muscles, sensory systems, and active behaviors would eventually emerge under almost any comparable planetary conditions. The existence of predators, locomotion, and ecological specialization may therefore represent an inevitable expression of energetic opportunity once sufficient oxygen became available. But inevitability only extends so far. The specific forms that survived were contingent on countless unstable details. A slightly different ocean circulation pattern could have favored different ecosystems. A more severe extinction interval might have erased chordates before vertebrates diversified. Another lineage entirely may have inherited ecological dominance. The Cambrian established the framework of complex life, but it did not guarantee which creatures would occupy that framework in the future. The ocean acted simultaneously as opportunity and restriction. Its calcium-rich chemistry encouraged the evolution of shells, teeth, and skeletal supports. Its oxygenated shallow waters made sustained swimming and active hunting biologically affordable. At the same time, the persistence of anoxic deep water created sharp vertical limits. Beneath the chemocline waited hydrogen sulfide-rich environments, hostile to most animal life. Diversity, therefore, compressed itself into relatively narrow habitable zones along continental shelves where light, oxygen, nutrients, and biological competition converged together. Every condition opened certain pathways while closing others. The Cambrian Sea did not merely contain life, it shaped the geometry of possibility within which life could operate. Even now, the continuity remains material as well as evolutionary. The calcium inside human bones once circulated through ancient seawater during intervals when trilobites crossed microbial sediment and radiodonts hunted through green water. The phosphorus forming the backbone of every strand of human DNA belonged to nutrient cycles already active in Cambrian oceans more than 500 million years ago. Water molecules passing through modern tides, storms, bloodstreams, and clouds moved through those ancient seas long before vertebrates existed. Nothing was preserved unchanged, yet nothing became entirely disconnected. The continuity between the Cambrian Ocean and the present world is physical, chemical, and biological at once. And perhaps that is the deepest perspective the Cambrian leaves behind. The ocean did not know what it was building. It possessed no awareness of mammals, forests, civilizations, or minds capable of reconstructing vanished ecosystems from compressed layers of stone. It simply existed within a particular arrangement of chemistry, geography, temperature, pressure, and time. But within those conditions, certain forms became possible. Predators emerged, nervous systems expanded, body plans diversified, extinction selected survivors unevenly across unstable environments. Evolution accumulated structures capable of movement, memory, perception, and eventually reflection itself. The future was not designed in the Cambrian Ocean, yet it was written there all the same, in the mineral-rich water, the crowded continental shelves, the selective pressures of predation, and the fragile creatures drifting through a 20-million-year interval that permanently altered the trajectory of life on Earth. The modern ocean still carries the architecture of the Cambrian inside it. Coral reefs crowded with fish, open water layered with drifting plankton and hunting predators, abyssal plains recycling dead organic matter through dense microbial communities. All of it follows ecological structures first assembled more than 500 million years ago. The names and species have changed almost beyond recognition, yet the underlying logic remains startlingly familiar. Primary producers capture sunlight near the surface. Consumers graze upon them. Predators move through the water column above. Decomposers return nutrients to circulation below. Energy passes upward through interconnected layers, while matter cycles continuously back downward into the sea. The Cambrian established this arrangement early, and nothing since has replaced it. Modern marine biodiversity, therefore, rests on foundations laid during an interval astonishingly brief compared to the rest of Earth history. Coral reef ecosystems now support approximately 1/4 of all marine species, despite occupying only a tiny fraction of ocean area. Vast pelagic food webs stretch across entire ocean basins. Deep sea ecosystems, invisible until recent decades, sustain communities around hydrothermal vents, methane seeps, and abyssal sediment plains. Yet beneath this immense diversity lies a relatively constrained set of anatomical frameworks inherited from the Cambrian world. The body plans remain the same. Arthropods still dominate animal diversity. Mollusks still occupy ecological roles from grazing to predation. Chordates still organize movement around the same notochord-derived architecture first visible in fragile Cambrian swimmers. Echinoderms continue building pentaradial bodies across seafloors. Cnidarians still drift through the water column using designs older than forests. The species multiplied, the architecture endured. This perspective changes the scale of the modern biodiversity crisis. Extinction has always existed, and species losses alone are not unprecedented within Earth history. What the Cambrian reveals, however, is that disparity, the existence of fundamentally different body plans, is far more delicate than diversity measured only in species numbers. Species may recover over millions of years through adaptive radiation and ecological replacement. Entire anatomical frameworks do not easily return once eliminated. No new major animal phyla have emerged since the Cambrian. The architectural possibilities established during that interval appear effectively fixed. Modern evolution modifies, elaborates, miniaturizes, enlarges, and specializes existing designs, but it does not generate entirely new foundational body organizations on the scale visible during the Cambrian Explosion. The ocean today therefore contains both extraordinary richness and profound limitation simultaneously. It possesses millions of species, yet nearly all belong to anatomical categories defined 500 million years ago. The Cambrian also survives at a more intimate scale. Human beings carry its inheritance physically. The bilateral symmetry of the human body, with sensory organs concentrated at the forward end and paired structures arranged along a central axis, emerged within Cambrian animals navigating directional movement and active predation. The segmental organization, visible in the vertebrate nervous system, reflects ancient developmental patterns already present in early chordates. Even the vertebrate eye, though radically transformed across evolutionary time, belongs to sensory lineages that intensified during the Cambrian expansion of vision and neural processing. The structure of the human body is therefore not separate from Cambrian history. It is Cambrian history continued. To stand upright on land while carrying a centralized nervous system, forward-facing sensory organs, a segmented musculature, and a mineralized skeleton is to embody anatomical solutions first assembled in ancient seas, crowded with trilobites, radiodonts, worms, drifting larvae, and fragile chordates moving weakly through green water. The continuity is deeper than metaphor. Every breath taken by a vertebrate, every movement coordinated by a spinal cord, every sensory signal processed by a centralized brain exists within frameworks shaped during that transformative interval. The Cambrian Ocean survives not as memory alone, but as biological structure carried continuously through time. And so the Cambrian never truly vanished. Its water still cycles through the modern ocean. Its ecological logic still organizes marine life. Its anatomical experiments still define the possibilities available to animals alive today. The sea that once held trilobites and radiodonts has altered chemically, geographically, and biologically almost beyond recognition. Yet its inheritance persists in every creature that swims through modern currents, crawls across sediment, flies through air, or walks across land. The Cambrian Ocean ended. And in another sense, it never ended at all. There are stretches of Earth history so long that entire mountain ranges rise and disappear within them. Oceans open and close, continents fracture, collide, and drift apart again. Atmospheres alter their chemistry. Ice advances across the planet and retreats. Extinction follows extinction, reshaping life repeatedly across spans almost impossible to imagine clearly. And yet, across all the time since the Cambrian Explosion, through approximately 485 million years of planetary change, no entirely new major animal body plan has appeared. The interval itself was brief, roughly 20 million years. Within that narrow window, the foundational architectures of animal life emerged with astonishing speed. Arthropods established segmented exoskeletons with jointed limbs. Chordates developed the notochord and dorsal nerve cord. Mollusks organized muscular bodies around radulae and soft locomotory surfaces. Echinoderms explored radial symmetry. Cnidarians refined drifting predation through tentacles and stinging cells. Annelids segmented themselves into flexible muscular units capable of burrowing and crawling. Everything that followed came afterward as variation. Evolution expanded these designs into forests, reefs, skies, deserts, deep oceans, and eventually cities filled with artificial light. Wings evolved, mammals diversified, dinosaurs dominated and disappeared. Primates emerged from tropical forests. Human beings learned to examine compressed stone and reconstruct vanished seas from traces preserved inside mountain rock. But the underlying anatomical framework remained Cambrian. The ocean that generated those frameworks no longer exists as a physical place. Its shallow continental shelves were folded upward by tectonic forces into limestone cliffs and shale deposits high above modern sea level. Sections of Cambrian seafloor were buried deep within Earth and destroyed through subduction. Ancient coastlines vanished entirely beneath younger sediment and younger oceans. The chemistry that once saturated Cambrian seawater with dissolved calcium altered gradually through hundreds of millions of years of biological activity. Even the water itself moved on. Cambrian seawater evaporated into clouds, froze into glaciers, flowed through rivers, passed through the cells of extinct animals, circulated through roots and bloodstreams and storm systems, and eventually returned again to the sea. The molecules themselves persisted, while every landscape surrounding them changed. The creatures are gone more completely still. Anomalocaris no longer moves through green water beneath sunlight scattered by algae. Hallucigenia no longer walks on soft lobopod limbs above sediment darkened by bacterial films. Opabinia no longer lifts its flexible proboscis through Cambrian currents while processing the world through 5 separate eyes. The Archaeocyath reefs collapsed hundreds of millions of years before the first tree appeared on land. Nothing alive has seen them. What remains are impressions in stone, compressed outlines preserved through improbable geological circumstance, and the consequences carried forward by survivors. The consequences endured. The structure of the vertebrate nervous system endured. Bilateral symmetry endured. The forward concentration of sensory organs endured. Eyes endured. Predation endured. Ecological layering endured. Complex food webs endured. The fundamental organization of animal life persisted through every extinction and every transformation that followed. And somewhere within that vanished ocean, moving through warm water rich with calcium and sulfur compounds, a small chordate swims weakly above the seafloor. Its body is soft. Its muscles contract in slow waves along a narrow notochord. It drifts past trilobites, crossing sediment with clicking legs and reflective calcite eyes. Shadows move through the water overhead, where larger predators patrol between layers of suspended life. The sea around it smells faintly metallic and mineral-rich, carrying dissolved chemistry through crowded continental shelves beneath unfamiliar stars. The animal possesses no awareness of future oceans or future continents. It does not know forests will exist. It does not know bones, feathers, language, cities, telescopes, or memory. It does not know that its body contains an anatomical possibility capable of surviving almost 500 million years of extinction, tectonics, climate change, and evolutionary transformation. It survives anyway, and now, across an almost incomprehensible distance of time, the nervous system shaped by that survival sits awake in darkness, listening to the history of an ocean that disappeared long ago and yet still exists within every breath, every movement, and every living creature descended from the world it made.